Audio. All right, I am seeing some audio on screen. Can you hear me now? Can you hear me now? We can't hear, still can't hear anything. Can you hear me? All right, I think I have an idea what is the problem. All right, just give me 1 second and I'll go over here. I think we're having a share in the stuff. Oh, so you can hear me now. If you can hear me, say yes. If you can hear me, say yes. Can you hear me? Yes. Okay, so we all can hear. Oh, I was of the opinion that we weren't hearing. Were you hearing the video that was being played? Yes, no? All right, so we're going to be having a fabulous evening. I did not come on at 5 because I wanted to cover something with you and I was not fully prepared to do so. Might I just say I'm going to spread on the screen in a bit the past papers that we're going to be covering between today and Sunday. We're going to be having a live tomorrow morning at 5. We're going to be looking at the 2017 paper today. And we're going to be, I'm going to tell you what exactly we're going to be doing right up until Monday morning before the exam. Now, I'm also going to be sharing with you some of my videos just to show you the animation because I'm not using a stylus. I'm using a touchpad, and it's not so easy for me to use the touchpad to do the drawing and all that bit. So I am definitely going to be showing you some of the things that I have to cover using videos. So I do hope that we are ready. I am trying to pull all the files that I have planned for you today. I got a new file today. So just let me go on over here. Everything was all set up, but I had to pull down the system because you said that you weren't hearing and all of that. All right, so we have the 2016 paper online already. So you could visit our playlist to get the 2016 for bio. HSB, we are working on you as well, but just give us a bit. So we are going to be sharing with you 2017, 2018, 2019, 2020, and 2021. We are going to be going through all those papers between now and Sunday evening. Now, we're going to be showing you some similarities between the paper. It's very important that we understand or we can pretty much the trend with these papers that you can pretty much figure out what the exam is going to be like. So like I said, we would have done the 2016 paper already. I am now pulling the files so I can just go through them very quickly with you. My son is up. Give me a minute. All right, Anai. All right, great. So that is pretty much a 6 past papers. All right, great. So, yeah, I'm sure. Yeah, great. Just give me 1 second, guys. Alrighty, thank you very much, guys. I know it's trying. For those persons who are with me, you know that I try my best to facilitate, but sometimes it's a little rough. So what I'm going to do before I start the show today, we're going to be looking at a series of papers that you can pretty much look at the trend. I don't know if everybody would have a copy of a paper, but we're going to try our best to show you the papers that we're going to be doing over the next couple of days. Oh, I hate to hear him cry. All right, so now we're ready. I'm going to be sharing the screen. So I'm going to be showing you the 2016 paper first for those persons who have never seen it. All right, great. So these are our papers. So this is a 2016 paper. I'm going to go through it slowly, and we're going to look at what is common over a 6 years period, a 6-year period. So here we go. So we're going through, so our first question here is pretty much on enzyme. It is a lab, and so you have this graph is to be done. And pretty much this question is pretty similar to the 2019 paper. So this is 2016. Just let me look over on 2019. All right, so look at that question. That was 2016. Look at the 2019 paper. Oh, no, not the 2019. Is it 2017? Sorry. That's 2020. Not 17. What's happening here? And I don't think it's 18. I didn't do 18. All right, look at 18. 18 comes with a graph, starts pretty much with a graph. All right, and this is an I. That's 17, I think. And this is 2021. This is a 2021 paper. And again, this paper has a graph. All right. So the paper we're going to be looking at this evening, just let me show that to you, being that is the big thing here. So we're looking at the 2017. So you would have seen the lineup 2017, 2018, 2019, 2020, 2021 are all the papers that we're going to be covering for you today. So I'm going to go through the question. I did sit and I worked these things out very quickly just now, and we're going to be looking at them, and you're going to be assisting me as we go through the paper. So just let me check here. I think I'm still getting used to this software. I said so, and the title, so I can. All right, so are we ready? So usually I start my show with a roll call, but the number is so large today, I don't know if I can call everybody's name. Now, just tell me where you are from in the chat, as we're going to be having a great time for the next 2 hours thereabout. I'm from Jamaica. I'm pretty much in Kingston, Jamaica. I like to know the reach of the channel. I know that we have a whole lot of persons out of Guyana and Trinidad, but it would be nice if you could just share with me wherever you are from in the Caribbean. You don't need to tell me exactly. I'm from Trinidad, I'm from Guyana, that type of thing. That would have been nice. HSB. Now, there's a misconception. As persons just tell me where they are from, there's a misconception about HSB and biology, where persons are thinking that if I am not doing HSB, then I shouldn't look at the revision for a bio paper. But remember, this is just a plant component except photosynthesis. That is going to be a real difference between the papers. So when there is a revision between bio and HSB, if you have nothing else to do, it would have been nice if you sit into the session and make sure that you understand a thing or two. Now, question number 1 in most cases is not going to be so suitable for the student doing HSB. However, all the other questions usually, in most cases, are going to be suitable for students doing HSB or biology. So I'm seeing persons from Guyana, I'm seeing persons from Jamaica, a lot of persons from Jamaica. I'm seeing persons from Trinidad. I think I see Antigua And I am also seeing, so I think I saw Antigua. Yes, I saw Antigua, Jamaican, a whole lot of Jamaican in the room. And we have the Guyanese. If you don't understand my accent, just let me know. All right, because sometimes I just break out and talk. I've seen St. Lucia as well. Welcome to the show. All right, so Jamdom, hi Jamaican. All right, so I'm seeing a lot of persons from all across the Caribbean. We know that we are in quite a number of countries, but it's always nice to know who we're talking to. Welcome to Jamaica. Welcome to Mr. Wilson Live. Welcome to CSEC Biology TCP. And as we're going to see scrolling at the bottom of the screen shortly, we have an online school for persons who want to be a part of a school come September. So without further ado, let me just, thank you. Let me just hop right into the paper. I'm going to take myself off the screen, or shouldn't I? Probably I shouldn't. So let me just hop on over to the paper and we move right on down. Where's the paper? Great. All right, so I might be doing a little reading for you. All right, so if you are doing the alternative paper for biology, that is the paper 032, it's very important that you take part in this session. It might be wise I take myself off the screen for you to see the paper a little better. And somebody is going to say, sir, you need to magnify the thing so I can see it a little better. I'm going to do that as well. All right, so we might not see a lot on the screen, but we're going to see everything. As soon as we get driving, get going, everything will be nice, so don't worry yourself too much. All right. All right, great. I'm ready to go, and so should you. So for the first question here, we are looking at the 2017 May-June Biology Paper 2, and we're required to look at all the questions here. So for section A, we're supposed to answer all questions in this section. You're supposed to write your answer in the space provided in this booklet. It's very important for you to have a notebook with you now and a pen and a pencil, and respectfully, you can, of course, refute our claim or what we will present today. But whatever you do, ensure that it is done in a respectful manner. We continue to say that we are not the keeper of knowledge, and we invite challenges from students and teachers at any time. That is how we grow, or that's how the scientific community grow. So at no point we are going to ask you not to comment. At no point we are going to ask you not to say, I don't think it is so. That is fine on this show. That's what the show is about. Now, whether your answer is right or not so right, it is good. I tend to like the not so right answers, because the not so right answers help those persons who thought that they had it right only to find out that it is not so right. There are usually some persons with some misconception, and when we have these misconceptions, they tend to move from year to year, from generation to generation, from one child to another. And we want to take the opportunity to correct them. So whatever you have to say, say it. Now, in this room, it is very important that you are empathetic. So you are not allowed to laugh at anyone, despite you are the brightest person on the planet. You are not allowed to laugh at those persons who are learning, because this room is pretty much for the persons who want to learn and those persons who want to share respectfully. So we're not going to be laughing at anyone. We're not going to be calling anybody an idiot. We are not going to be flooding the room with comment that doesn't make any sense. We are going to be respectful of each other. If we violate the rule, then I will have no option than to protect the other persons who would love for them the space to be protected. All right, so here we go right into the paper. So we're expected in this experiment, some students put corn seeds into each of two test tubes labeled A and B. Now, what I want you to do, I want you to look at the experiment on the screen, and I want you to type in the chat for me what experiment you think these kids were doing. If you observe here, we have moist cotton, And of course, we have the seeds in that moist environment. What do you think that they were testing? What type of experiment they were carrying out? Tell me in the chat, because it's going to be a real interactive session. What type of experiment they were carrying out? Or they are carrying out? Tell me in the chat. Great. So as you try to think about what is happening here, because it's not stated, let us move on to the first question. Now, you're supposed to write a suitable aim for this experiment. A suitable aim for the experiment. So I want you to quickly type in the chat, what is your aim for this particular experiment? And all the information that you need to write the aim is on the screen now. Now, please be reminded that the aim is pretty much what you intend to do. Indeed, it's germination. Very well. We're almost ready for the exam. It is germination. Now, I want the aim. What's the aim for this experiment? So the aim usually begins with 2. It is pretty much what you want to find out. What do you think the kids wanted to find out in this experiment? Yes, they were carrying out germination, but what they wanted to find out about germination? They wanted to find out the effect of temperature on germination. But for those persons who don't know what they wanted to find out and don't understand why Walbrook would have said that, you want to tell us why you think it's temperature that they were testing? Or they wanted to find out the effect of temperature? Just tell us why. Temperature, the effect of temperature on the germination process. Great, Bell. That's wonderful. There are some words I can't say. I can't even breathe to say. Casanova. All right. Great. Yes, it was temperature. But why temperature? What's the clue there that would have informed you that it is indeed temperature? What is there for us to see? Because of the 2 different temperature. Great. So it's right in front of us. If we were supposed to look, I'm sorry. If we were supposed to look at the top of the tubes here, you would have realized the difference in temperature, 10 degrees Celsius versus 20 degrees Celsius. All right, that's very important. Let us move on down. For those who are new, we usually have a lot of bikes passing by. We do apologize for that. Now, let's look at question number 2. In which of the following, in which of the tubes, A or B, would germination most likely occur? Give one reason for your answer. So we're looking at B now. In which of the tube here, A or B, would germination most likely occur? And you are supposed to give us a reason for your answer. So we're looking at 2 tubes. One of the tube, A, is, I think it has a temperature of 20 degrees Celsius, and B has a temperature of 10 degrees Celsius. Now we want to find out which of the tube is it more likely that germination will occur in. Persons are saying A, persons are saying B, persons are saying 20 degrees. And because you're a little all over the place with your answer, now you have to tell us why you think it is this tube. Validate your answer for us. Why is it A or why is it B? I don't know, so I'm asking you. So B is closer to room temperature. B is closer. B is actually 10, you know. B is actually 10. Just let me show you it on the screen again for those persons who might not be making the right choice here. Just let me show it to you on screen. So there we have the temperature. A, 20 degrees Celsius. B, 10 degrees Celsius. Now, why is it A or why is it B? The truth is, I don't know which it is. So I am relying on you to tell me the truth. All right. So, oh, I forgot. So persons are saying A, it needs a warm temperature. Thanks, sir. A. All right. Israel is saying A. All right, great. So I'm going to accept that it is A. It is A. Great. Now I want supporting evidence for A. I want concrete evidence. Why A and not B? Why 20 degrees Celsius and not 10 degrees Celsius? How would 10 degrees Celsius affect the seed? Germination is promoted at a higher temperature. All right, I am seeing that. A, because it has the temperature closer to the temperature suitable for the germination process. All right, I am not going to echo you. So I am going to ask for what I want. You're telling me now why it is A, and that's beautiful. Your answer is fine. I want you to now tell me why not B. So you have told me why A, and you're all correct. But why not B? Why not B? So yes, your answer about the temperature and room temperature and the higher temperature, yes, you're all correct. But I'm still not convinced that you understand why not B. Why not 10 degrees Celsius? Low temperature will slow the rate of germination. Okay, I'm looking for that word that Brown Nelson has. These temperature would inactivate the enzymes. All right, great. That's the answer that I'm looking for. I'm not going to prod you once I see it somewhere. So if the temperature is too low, then that will pretty much deactivate the enzyme, or it will pretty much cause the enzyme not to work. While we talk about enzyme, let us be reminded that enzyme is not an organism. Enzyme is pretty much a chemical. All right. So enzyme cannot be killed. We say that it is denatured or deactivated or something like that, but enzyme can't be killed. So the answer for that, it is because the lower temperature is, of course, going to affect the enzyme, cause the enzyme not to be able to function properly. Great, guys, you're doing well so far. Now, state two roles that the moisture from the cotton wool plays in the process of germination. Now, be reminded here that the moisture is pretty much water. It is not pyrogallic acid, right? It is water that is in the cotton. Tell me two roles that this would play in our process of germination. Two roles. All right, I like the start that we have so far. We're doing well. It means that we are going to be able to cover all the 6 past papers we want to do. All right, great, exactly. The bio exam is on Monday, so my focus is on biology now, but we are going to be having live right up to Tuesday night, Wednesday morning for HSB. I think HSB is on Wednesday. So we have you in mind, but just be mindful of the guys who have exam on Monday. We're going to try our best. And we don't want the students who are doing HSB to leave, because as soon as we move away from question number 1, we are going to be featuring content from the HSB syllabus as well. All right. Water helps to split the tester. That's a good move. All right. So guess what? Anna is here, and look at Anna. Anna is down with his measuring cylinder. Yes. Say hi. All right. So Anna is down with his measuring cylinder. He was given a little trouble just now. He's usually here. He's my co-teacher. In a matter of fact, he thinks that he's a better teacher than I am. All right. So let us look. The water is absorbed through the micropyle to allow the water in inhibition stage, okay, responsible for the digestion of nutrients in the seed. Digestion, I've never heard it like that before. Causes water to enter the seed and pretty much causes swelling. All right, I'm hearing about the swelling. I'm hearing about the splitting of a micropyle, but remember that water is also needed to activate the enzymes. All right, and water is also needed, as somebody say about digestion. I wouldn't say digestion, but water is, of course, needed for the release of that energy from the seed there. So we want to ensure that we have a good grasp of that content going through. So we want the glucose from the seed here for the process of respiration, and water is, of course, going to stimulate that reaction. So moisture provides the seed with water, which is important for the metabolic reaction and activate the enzyme. I think that is perfectly said, and I think I like that. All right, great, guys. So we're going to be moving on with that. I think we're getting some real good answer here. So we look at the other question. It says here, state two roles. Oh, that's what we did just now. All right, so we're going to be moving on to the other question. So this is the other question. Now, remember that question number 1 on the biology paper is your lab question. So we're looking at some words that will be featured or can be featured on the question number 1, words that you should go to the exam armed with. Now, some of the words that you want to harm yourself with going to the exam, there are going to be words like aim, hypothesis, precaution, control, apparatus, material. And just before we move on, just remind me in the chat, what is the difference between apparatus and material? And as I go through the session, I'm going to be pulling on some of those lab knowledge so that we can master question number 1. So tell me in the chat, what is the difference between material and apparatus? What is the difference between material and apparatus? I'm upping on over to the question as you type that for me in the chat. So we're looking at this question, and at a glance, we're seeing that this has to do with phototropism, the response of plant to light. So it said that the corn seed which germinated are placed in a pot and exposed to unidirectional light as shown in this figure. So unidirectional there means that the light is coming from one direction, from one side. Just pretty much show a peephole as we're seeing here. No, don't do that. As we are seeing here. Now, let us look at what the question wants us to do. It says, what type of movement is shown by the plant? Now, if you look at the plant there, the plant is definitely showing some movement because you're seeing the plant moving here, moving in this direction if you're looking at the screen. Tell me in the chat there very quickly, what type of movement are we witnessing there with the plant? So apparatus are pretty much an instrument. Apparatus can be reused in the lab, like the measuring cylinder. It is a apparatus. And we have some more over here. We have the tripod stand. We have funnel beaker. All of those are examples of apparatus. Now, when we think about material, we're thinking about things like filter paper. Reagent could pretty much fall pretty much in that category, but usually we keep reagent by itself. If we're talking about the splint being used, that's another one. If you are using napkin in the lab, anything that is, should I say, expendable, anything that you will have to throw away after the lab, that is pretty much considered to be material. So we are seeing Gnostic movement. Interesting. We are seeing phototropism, growth movement. And all right, so I am seeing something here, and I like what is happening here. I want the person, let me see, Gnostic movement. Could you just define Gnostic movement for us? You could just up on over to Google and just define Gnostic movement. It is a nice word and probably a word that everybody should be exposed to. So I'm going to ask you to just define that there for me in the chat. What's Gnostic movement? So we are seeing phototropism. All right, we're seeing a lot of nice words there. We're seeing phototropism. We're also seeing plant axin, right? I'm not too sure where that is going. I'm seeing growth movement, and I'm seeing growth movement being likened to phototropism. I'm seeing persons coming in with material, and they're saying that material are pretty much instrument you will be using to carry out the experiment. No. I just gave that definition for material. We don't want to say instrument, and we don't want to use this word loosely because CXC is pretty much knowledgeable of the fact that you really don't know the difference. So Gnostic movement is the movement of a part as a result of external stimulus. This is interesting. So when we talk about Gnostic movement, we are looking at things like when you touch the shame old lady. So the shame old lady, based on the change in the environment, you would have touched the shame old lady. It is moving. I know that this would have caused some controversy. Now, Gnostic movement, plant movement that are in response to a stimulus, I think. All right, it is indeed in response to a stimulus. Now I want you to find examples of that stimulus. This is student-centered learning, so I am going to be prompting you to get that answer. I don't want to tell you what is wrong or right, whereas when we have some controversy, I want to hear two sides of the story. So is it growth movement or is it Gnostic movement? Which it is. So you could even ask Google what it is. The response of a plant to light, is it growth movement or Gnostic movement? I'm not too sure why we're saying sunlight. Phototropism. Phototropism is pretty much a response of a plant to light, and we have geotropism, where the plant is responding to water. So, temperature. I think some persons are coming in late. I'm sorry, some persons are posting late. So Gnostic, because it responds to light. Interesting. All right, let me see this. Touch the shame old lady plant, mimosa plant, is an example of Gnostic movement. Great. So touching the shame old lady and the shame old lady closing, that is an example of Gnostic movement. But the movement of the plant to sunlight, is it Gnostic movement or is it growth movement? That's what we want to find out. You want to go look for mommy? All right, peekaboo. Is it growth movement or is it Gnostic movement? And now that we have opened this new can of worm, that's exactly what CXC wanted to find out. Is it Gnostic movement or is it growth movement? And if it is growth movement, why can't be considered to be Gnostic movement? Can somebody clear that up for me, please? If it is growth movement, why can't it be considered as Gnostic movement? What? What? If it is growth movement, why can't it be considered as Gnostic movement? I just want somebody to help me here. All right, I am listening. I know that the Gnostic movement is most time used to look at the Venus flytrap and the shame old lady. All right, but we're hearing persons saying that what we have here, it is in the Gnostic movement, and we want persons to just defend the point, right? We really don't want to say that you are wrong or you are right. Okay, it is responding to light and not touch. All right, there we go. Can it be considered both? Interesting. Is it not Gnostic because it is caused by external stimulus? I could open this can a little wider, but I really don't want to go there this evening. Opposed to the plant internal structure. All right. But the plant is always growing in relation to something. All right. It's always moving in relation to something. It's moving towards the that missed me there. It don't respond to touch. Gnostic movement is response to stimulus such as touch. I don't know. All right, somebody can just know for me, please. Is it movement towards a stimulus or it's not Gnostic? All right, I am going to be. I think it can be classified as Gnostic movement because no single part of a plant is reacting immediately after being. Let me just read it. I think I like where we're going here. And for those persons who are a little impatient, you have to be with us because we want to ensure that we are removing things that shouldn't be. All right. So reacting immediately after being exposed to a stimulus. All right. Gnostic movement means stimulus is touch, and it is reaction of a plant to touch, while growth movement is plant movement upward. All right, so tropic movement depends on the direction of the stimulus. It's always going to be to a stimulus. All right, so because it's permanently growing in a particular direction towards the external stimulus, whereas Gnostic movement would be responding to touch. All right, it grows towards the light, not away, so it would be growth. All right, my brain. I'm liking it. I'm liking it. I'll soon give you the answer. All right. Let me see what else is here. Gnostic movement are non-directional responses. This sort of sound good. Gnostic movement are non-directional responses to stimulus. Example, temperature, humidity, light, iridescence, and are usually associated with plant. All right. Now, having looked at this definition and the one I read before, now what is our position? These are two nice definitions. It can be growth because growth takes place. It's not takes place. Let me see there. It is not, okay, there's somebody coming out now and finally speaking. So learning start to take place now because at least somebody come out and take a position. All right, just bear with me. We want to Gnostic. All right. I don't do bio, just guessing. Okay, that's fine. It's all right. Gnostic movement and okay, I think we look at that already. All right, it can't be growth because growth takes days, I think. All right, no, growth doesn't really take days. You're growing or things are growing, but really you don't see it unless you have it beside something. It's not so easily seen. It's not Gnostic movement. If it was Gnostic movement, it would not grow in the direction of light. So I have it as growth movement. All right. And I do go with those persons who are saying that Gnostic movement is like to touch or to what we refer to as the stimulus outside of the sunlight. Because the plant, you're going to see the question that comes next, where we are going to change the position of the light and the plant is going to grow. And this is a real phenomenon in the environment where we are seeing plants falling and then shortly thereafter they are growing. So for me, and to my best knowledge, this is of course going to be a growth movement and not Gnostic movement. But of course, if you have something that you can convince me otherwise, as I like to say, I would be very happy for you to share that content with me. I think we might have some literature on movement. I'm just going to ask my assistant to just check that for me. But as far as I know, it is growth movement. But where there's controversy, we're going to have it all sorted out. Do me a favor now. Gnostic movement for me. There's a controversy around what? No, Gnostic movement is not in the textbook. All right, but I have seen Gnostic movement, and I think TBJ brought Gnostic movement as well. I saw it there. All right, so Gnostic, all right, Gnostic movement, according to Google, means the direction of response. Let me see. Means the direction of the response is independent of stimulus, but in the case the plant is moving direction towards the stimulus. All right, so like I said, my understanding is that Gnostic movement is pretty much something that is synonymous with a change in the environment. Usually, like if you touch the plant or something like that, it would be more considered as Gnostic movement. Now, usually we say that there are two types of plant movement. Our group. Turtle are growth. All right, hold on, guys. There are really two types of plant movement. And the two types of plant movement are either going to be tropism where the plant is growing towards light, where we call phototropism, or geotropism, where the plant is growing towards water, gravity, and then there is Gnostic movement, as when you would have touched the Venus flytrap and it closes, or you would have touched the mimosa plant and it closes. That is my knowledge on the content, and we move on in the lesson. But if you so feel to continue with this topic, please send me an email and we can talk. So for me, this is going to be tropism, phototropism. The plant is growing towards the light, as is the norm in the environment. So for this, I give it the answer as a growth movement, and the particular growth movement is phototropism. So it's growth movement for me. All right, let's move on to the other question. State the hypothesis that the student could test by using the apparatus and material shown in figure 2. By using the material, the setup in figure 2. What is a good hypothesis for that? A good hypothesis for the setup in figure 2, what could we do? What's the hypothesis that we could have? All right, guys, please be reminded, if you are not in for my answer, don't be afraid. Consult with your teacher. Go to the back end of my YouTube channel or on any video. Send me whatever you think it is, and I will continue. That's how I learn. That's how I grow. All right? But for now, that is what I know, and I'm sharing that with you. But I'm open for new ideas. All right, the plant will grow. I think I said something. The plant will grow in the direction of light. All right, Peter Gay Campbell. Yes, I agree. Peter Gay, plant grow towards direction of light. Plant will respond positively to light. That thing there that says that the plant will respond positively to light, it is so wide for a research. What is really positive? So for the others that I have read so far, yes, I like them. Growth is affected by the direction of light. Growth is affected by the direction of light. That can be tested, but I don't think that that would have been the most ideal hypothesis for this experiment. All right, the shoot of a corn seed grow in the direction of the light. Now, it wouldn't be a specific thing as to corn. We could talk about the shoot of a plant, because all plants pretty much with the shoot would have been behaving pretty much the same way with whatever light. So I am seeing some nice ones though, and one of the things about an hypothesis for an experiment, not everyone will have the same hypothesis. But usually the hypothesis is something that speaks definitively to what you think something is. So if, for example, let me just use something that is way out of the book. Suppose we all thought that the sky was red. So we all look at the sky, and some persons will say that the sky is red, the sky is blue, the sky is whatever color. And then we could carry out an experiment to check that wavelength, that light in there, and then we could determine whether it is right or not right. Please be reminded, an hypothesis does not mean that your views are true. If they were true, if they were already known, then there would be no need to test. So the hypothesis that we have here, that the plant will grow towards light, anything around that is pretty much a good hypothesis, because if we look, we are using this picture to create the hypothesis, and what we're seeing here is that the plant is growing towards light. So that is pretty much anything along this line is going to be a nice answer. And I think I like where we're going. I think I like the class today. A lot of persons are pretty patient. And I just take your time. All right, apparatus material. I lost my bearings now, guys. Okay, great. I'm right here. So we're moving on to the other question, which said that students want the ceiling to grow straight. Now we are going to talk about, now you are going to now have an opportunity to go back into your memory as to whether it is Gnostic movement or growth movement. Look at this particular question. It is saying the student wants the ceiling to grow straight towards the ceiling. Suggest how they could manipulate the experiment to get this result. Tell me in the chat, how could they manipulate the experiment to get this particular result? So they want the plant now not to grow towards the light, but instead to grow towards the ceiling. How could they manipulate the experiment to get that sort of a result? You are watching Mr. Wilson live. If you have not yet, please be reminded to like, share, and subscribe if you like the content that we have here. We have approximately 300 videos on the channel surrounding biology and HSB. Please ensure that you access them at your leisure. There are 2 playlists on the channel, one for biology, one for HSB that has a series of past papers, paper 2 and multiple choice. You want to ensure that you endow before the exam. We're going to be with you until the exam, after which we are going to be moving over to HSB to have some live sessions with the HSB students who will be having their exam, I think, on Wednesday. So we are looking here at what could be done to have the plant growing straight towards the ceiling. So put the light above it. Great. They could place the light source above the plant. They can change the direction of the opening of a box as well as the lamp. So we're getting some interesting finding here. And that is why I'm saying one thing I like about science is that different scientists will come up with different ways of solving the problem. One thing is that some of the ways are going to be more efficient than others. For example, all plants, in most cases, all plants will be growing towards the sky. So if we move the light from one side, and we put it to another side, of course the plant will bend and the bending will continue. But what we want to do is to get the plant to grow upward. I remember we said that it was growth movement, preferably phototropism, where the plant is moving towards light. So of course, if we were supposed to put the light source above the plant, then the plant would be growing towards the ceiling. So anything around that, putting the light above the plant, would have been a good answer. I'm sorry, I can't look at all the answer. The screen is really a buzz. So I tend to just give the general answer and you can write them in as you go along. If you are using a computer and you can print screen as you go along, you can do that. But we're going to try as best as possible to make this paper available with the answers by sometime tomorrow. All right, don't hold us to it, but we're going to try our best because we have to put out the others. All right, so yes, put the light above it. Let's move on. The ceiling shown in figure 2 use all its nutrients stored in the cotyledon. Discuss the process by which these plants can make more nutrients. So we know that stockpiled in the cotyledon was a whole lot of food storage to enhance growth. Now, the plant would have exhausted all that nutrient stock, and the plant now wants to get some more energy, some more nutrients in order to grow. What is it that they are going to do? What is it that is required by the plant now that it has expended all the energy stored in the cotyledon? What is it that the plant needs to do? Let me go back to that question and read it quickly for you. The ceiling shown in figure 2 use all the nutrients stored in the cotyledon. Discuss the process. Please pay attention to the word process. You want to now discuss this process. It is a process by which these plants can make more nutrient. Now it is a plant. It is no longer a seed. How is it? Okay, I am seeing the answers popping up on the screen. Beautiful. So I am seeing photosynthesis coming up. And that is indeed the process that we are looking for. Now, photosynthesis, let us see what else will be required. Wonderful. Not hearing. Oh, persons are hearing, but some persons are not hearing. I'm not too sure what's happening. All right, great. So the process there is, of course, going to be photosynthesis. No, 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 not watering the seed. The process is going to be photosynthesis. But let us look to see if we will have answered the question. Here it says that we are supposed to discuss. So what we have done by just saying photosynthesis is to name the process. So now that we have named the process, what we want to do is now to discuss what happened. So here you are going to tell me, nicely done, I'm seeing girl putting in something there, photosynthesis, and she's giving that chemical equation, which is of course balanced. She's saying 6 carbon dioxide plus 6 H2O is going to give you C6H12O6 plus 6 oxygen. Please be reminded, in the exam, if you are going to be writing this, you need to ensure that it is balanced. I observe, however, that you would have left off sunlight and chlorophyll. You really don't want to leave that out of the equation, because if you do, then it is no longer what you intended to do. All right, so plants can make more nutrients by photosynthesis. The plant utilizes light energy from the sun, water from the soil, and carbon dioxide from the air to make glucose and oxygen. That is nicely said, but I also like to see my equation when it comes to photosynthesis. I really want to see you practicing photosynthesis. And I know we have a drawn out session here, but I have to be doing the past paper and teaching at the same time. So that nice answer I saw just now is pretty much a nice answer, discussing what is happening there. Let us look at how much mark we would have garnered for answering that question. So it would have been 4 marks, so that would have been adequate. But to make it a little nicer and spicy, it would have been nice for you to write that chemical equation or worded equation to show us or to summarize what is happening with the equation. Now, there is something I wanted to look at. I hope everyone, just let me answer very quickly before I move on. Do you understand that which girl did? Do you understand that which girl did a while ago? Be careful the light and dark stage. I don't think you're using that so well. So let that stay a little bit. Because when you start to go there, it start to complicate the thing. Yeah, we understand that, but we don't want to go there because it might just push us over in the Calvin cycle. All right, hold on. Do we understand how to balance the equation? Because I know there are a lot of students here who are doing biology or also doing chemistry. So if you look at photosynthesis, yes, sir. If you look at the equation that she has there, you would have realized that she has the same amount for each of the element. Now, the easy way, I'm not too sure how your teacher taught you, but an easy way is just to split it down the line like that. So this would have been the line there, and I think you have C, H, O, and you want to ensure that the same amount of CHO that is on that side, it is pretty much the same amount on this side. So usually we use this line separating and try to ensure that what is over here, it's the same amount over here, and we add it up and so on. All right, great. I'll do the multiplication, whichever you were taught. So let's move on to the other question, being that you would have mastered this question. The other question is going to be, discuss the process by which raw materials required for making nutrients reach the leaf of the plant. So here you would have seen the equation that was written by girl. Now we need to use that equation to answer this particular question. Discuss the process by which raw material required for making nutrient reach the leaf of a plant. So tell me what happens there. I am very, very interested, and I'm looking for some real nice words. Oh, you made a song to remember it. Great. We want you to share that song. Send us that song so we can put it on our channel so other persons can learn from what you would have done. Send the lyrics. Oh, so that means that person's, all right, if you can't balance the equation, I'll probably just do a video and share with you. I know you're going to be a nice boy, all right? Great. Peekaboo. Right. All right, so we want to know, so osmosis and diffusion and transpiration. Yes, you have some nice key term there, but you really don't answer the question. Because if you're just writing those three names, all that you're doing is to identify the processes. But are you really telling us about it? And it says, just let me just go down. It says discuss. So to just name the three processes there, really would not garner the mark that you are looking for, because you have to pay attention to the fact that the question is saying that you should discuss. You should discuss, and you didn't discuss there. So you want to know, now that you have your key points, you have just written those to the side of your exam paper. Now you want to ensure that you now can elaborate what happens with osmosis, diffusion, transpiration, pull, because I don't know what would have happened in osmosis. I don't know what would have happened with diffusion. I don't even know what is transpiration pull. I don't know what is turgor pressure. I don't know what is the osmotic gradient. I really don't know any of those stuff. So I'd prefer for you to write it out exactly what happens for those persons who don't know, so they could learn. And the xylem is a wonderful name being mentioned there. Wonderful name. All right. Water reaches the plant by entering the root here of a plant by osmosis. It then travels up the plant through the xylem up to the stem, to the leaf. But yes, but yes, what about those? Yes, the water now gets into the root. That's fine, osmosis. But how does the water move from the root upward through the plant? There must be a process that is responsible for moving this water. Now that it's in the root, we want to move it from the root up to the leaf. So we can't just leave it there to say that it is actually osmosis taking it into the root. Yes, that is nicely said. But now we need to move it from the root to the leaf. What process is responsible for that? And one person said earlier, the transpirational pull. So the transpirational pull, what it speaks to, I think we spoke about something like that last night. What it speaks to is the pull of the water. We spoke about cohesion, where the water molecule, the water molecules, they are pretty much stick to each other, stuck to each other, touching each other. And as this one move away, what happens is that it is pulling this one with it. So we have this chain, and this is just pulling, and then we have this continuous flow. So transpiration pull is, of course, one of that process that is responsible for moving this water from the root up to the leaf, and then it gets out of the leaf, evaporation takes it into the environment. So osmotic pressure, the osmotic gradient, will move it from the soil into the root, and from the root we have the turgor pressure. We have so many stuff. We want to stay with transpiration pull, would have pulled that water up the column. Now, translocation is pretty much a movement of whether it be water or the nutrient via the phloem vessel. Translocation, in some cases, I've seen it where it is used to refer to one. I've seen it where it is used to refer to two. All right. Continuation. Water is, okay, great. Water is then evaporated through the leaf by transpiration. But yes, it goes through the leaf, but you also need to tell us about that pull. We want to know how the water moves from that root up to the leaf. So we want to talk about this transpiration pull, and we talk about the turgor pressure, we talk about the cohesion, all of these fancy stuff you could look at if you so desire. And of course, please be reminded that it is a xylem vessel that is taking the water from the soil. Being that we're talking about xylem vessel, we're doing past paper, and we're teaching at the same time, please let me just highlight something here. Now, we could talk about, once we talk about the xylem, it is also expected that we're going to be talking about the phloem. Now, let us talk a little bit about the structural difference. When we talk about structural difference, we're pretty much talking about the makeup. So the xylem vessels, they are dead. The phloem vessel, they are alive. The xylem vessel, they have lost their sieve plate. They don't have any sieve tube. However, the phloem vessel, they have sieve tube. Now, the xylem vessel, they are really tough. They are highly lignified, and the xylem vessel, they have no companion cell. However, the phloem vessel they have companion cell, and the companion cell has a nucleus. It is not the phloem vessel that has a nucleus. It is the companion cell. Great. Now, that is the structural difference. But there's also functional difference when we look at xylem versus phloem, in that the xylem transport water and the mineral salt up from the soil, while the phloem vessel would have been transporting that manufactured food from the leaf. We touched a lot there just now. I do hope that we can recall all that was said just now, or you could just watch a replay. All right, let's move right along. You'll find that the paper is getting a little more difficult as we dive deeper into the content. Now, let's look at this. This is another lab question, and we are now supposed to do a starch test. Starch is the most common carbohydrate that is found in leaves of green plants. Describe a test that the student could do in the laboratory to investigate whether starch is present in the leaf or in the leaf of a seedling. So let me see your starch test now. I think everybody would have done that for photosynthesis. You would have done one of the starch tests. And while you concentrate, we are multitasking. So just let me talk about those labs for photosynthesis. There are a number of labs for photosynthesis, and the lab would include like the evolution of oxygen, where we use the Eloda plant in the water. That is one lab. And if oxygen is present when we put that funnel over the Eloda and that test tube over the funnel, if oxygen is collected in that tube and we are supposed to properly secure it, then the oxygen will relight a glowing splint. That is one test, and that is a test for oxygen. It will relight a glowing splint. All right? No, I don't want iodine. I want more. I want the entire test. Next lab. Now, there's also the test where we want to determine whether or not chlorophyll is important for the process of photosynthesis. In this particular lab, we are going to be using a variegated leaf. And a variegated leaf means that's a leaf with more than one color, usually green and white. Now, all these experiments are on my channel for those persons who are doing the alternative paper. So we looked at the oxygen, we looked at the chlorophyll. There's another thing that we could look at. There's one that we look at carbon dioxide as well. We could also look at the euglena, its response to light. But let me see what is happening here with the test. When we are testing for starch. Let me see what the question says. It says that we are supposed to describe the test. Just to write the word iodine test is not adequate to describe the test. So you really don't want to get to the exam and just write in iodine. I'm seeing Casanova making an attempt here. Let me read what Casanova has. It says here, place a leaf into a beaker of boiling water. Great. To break down the cell membrane and the cell wall. Great. And the cell wall. 2. Turn off the Bunsen burner. Turn off the Bunsen burner and place a leaf in a test tube containing ethanol to remove the chlorophyll. All right, great. I hope when you were writing your procedure, you did not include what it does. All right, Peter gave, but that is incomplete. So I am waiting for the other part. So let me look at somebody else to see what happens. All right. Oil the leaf, then place it on a white tile. What? Who said that? I am seeing 3, so that means you had 1 and 2. So let me look at what 1 and 2 was. All right, I'm seeing 2. So 1, boil the leaf in water until it is soft. 2, boil the leaf in ethanol to decolorize. Remember, this ethanol thing, you are supposed to ensure that the leaf is placed in this boiling tube in the ethanol, and the ethanol is supposed to be placed in a water bath. Please be reminded that the ethanol boils at 78 degrees Celsius, water boils at 100 degrees Celsius. So if you were supposed to put the ethanol over open flame, you are going to have an explosion. But I have never seen in any test that I have done where the leaf is supposed to be oiled. So this is sort of new to me. So let me see. You are saying here, oil the leaf, then place it on a white tile. Cover the leaf with, I have never seen oil the leaf. That is something new to me. All right. I don't know why you would. Probably you could explain that to me. I have never seen it even in a textbook. I'm very sorry. I don't know if it's an error. So after the leaf would have been removed from the ethanol, the leaf would have been placed into it would have rinsed it in a water bath because the leaf would have been crispy, having been removed from the ethanol. So to soften the leaf, you'd place it in some water. Then you'd place the leaf on a white tile, after which you are going to add iodine to the leaf. Now, when you add the iodine to the leaf, you are going to note the color change. So I'm seeing somebody here saying note the color change. And I think the person is saying if the color changed to blue-black, then of course starch is present. So that is pretty much a test for starch in a leaf. That's what you want to have something like that written on the paper. Now name one plant organ other than leaves where starch may be found. One plant organ other than the leaf where starch may be found. One plant organ other than the leaf where, guys, remember we don't laugh at anyone here. Guys, we don't laugh at anyone here. Even if it is funny, keep it to your little self, but please do not laugh at anyone. There are persons here with different practices. Some of these are some malpractice, and they're a part of this room to learn from the persons who are laughing. So please do not laugh at each other. All right? That is violation of the rules in this room. All right, so we're having stem and root. Great, and seed. Now, with that said, I cannot move on without clearing this up. A yam is an underground stem. I want you to research, go to Google, and I want you to find out which is a stem tuber and which is a root tuber. You want to look at Irish potato versus sweet potato. Which is a stem tuber, which is a root tuber. Do that for me. Type it in the chat as we move to the other question. So all those that you gave just now, they are correct. Now, instead of starch, animals store complex carbohydrates such as glycogen. Name two organs in an animal's body which store glycogen. Two organs in the animal's body that store glycogen. All right, and there we are going to come up with another little bit of teaching there. Yeah, we have a whole lot of yang bike. I tell you, I'm so sorry, guys, but there's not much that I can do. Good night and welcome to the show. All right, great. So I wanted to find out which is, I have seen it on the biology paper, I think on the multiple choice paper, and a lot of students can't tell what is the difference between root tuber versus stem tuber. Yes, the liver is one of the answer, but what is the other answer? The liver, yes, the liver is right, and the muscle is also right, crossing over. Yes, we are doing well. So it's going to be the muscle and the liver cell where the glycogen or the excess glucose is converted and pretty much stored as glycogen. There we would have completed 25 marks for question number 1. Question number 1 is marked out of 25, and I think most persons did well just now, and I am so grateful. I think I like the response that we have been getting so far. We're going to be moving on into question number 2, but just before we hop on into question number 2, I must remind us that come August, the end of August, we are going to be making available my new book. My new book, I have 3 books coming out by September. We're also going to be having a series of activity book for some of the sciences. So you want to look out for that. It will either say Ricardus, author Ricardus Wilson. We're going to talk about that. We're going to make it public as we go along. We want you to support the brand. The brand will be on all major platforms. All major platforms that you can find a book to purchase, you are going to be finding that there. We want to also make known of our paid platform, TCP Academy, for those persons who are going to be sitting the exam next year and you are going to need some help. Please sign up over there. We're going to be there to offer you animation and all the help that is needed. Okay, root tuber, potato, and stem tuber, sweet potato. Really? So root tuber, what is the source of your information? Where do you get that information? From what site? Yes, so we are doing the Biology 2017 paper with a hope that we're going to be having another paper tomorrow morning at 5 o'clock. So we're going to go live 5 o'clock tomorrow, either showing you videos or we're going to be showing you another past paper tomorrow. But we have, I think, 6 past papers to complete by Sunday evening. Let's move on. The main difference between stem tuber and root tuber is that the stem tuber is a swollen stem, whereas the root tuber is a swollen root. Great. So give me an example now. So root tuber is going to be a sweet potato, and stem tuber is going to be so there we have the controversy. So some persons are saying that the stem tuber is this and the root tuber is that. That is something to challenge. The agriculture students, I want you to share with us because I'm sure it is captured nicely in your agriculture textbook. We're going to get back to that sometime throughout the show. Let's look at question number 2, a question that I really like. Now, CXC, if you ask me, is pretty fair a lot of the times. If you look in the question and you search, sometimes you can find the answers. And this is one of the cases where we can find all or most of the answers here. Question number 2, figure 3 represents the Whittaker 5-kingdom classification of living things. Remember, our kingdoms are, I'm not going to go into this as yet, just remind us of the kingdom in the chat. King Pharaoh called only female generals softly. Write them in the chat for me. The kingdom, we want to remind ourselves of the kingdoms. All persons are answering the questions. All right. Okay, great. So as we think about the kingdom, let me just follow your lead here. All right, so great. So if we're supposed to answer 1, then if we're going to map, a boy is mapped to a girl. So a man to a woman, left to right, up to down. So prokaryote to eukaryote. Now, eukaryote, if you can't remember the difference between prokaryotic organism and eukaryotic organism are you and I. We tend to have our nucleus membrane bound, while prokaryotic organism, they really don't have this nuclear bound nucleus. If you think about like the bacteria, it has or the bacterium, it has a nucleotide or a nuclide. I can't even remember how to pronounce or call it, right? But it is not bounded by a membrane. So our answer for 1, of course, is going to be prokaryote. Now, if we look at 2, let's move on over there, and the clue for 1 is right below 1. If we come on down here and look at the flow diagram here, like a dichotomous key, it says eukaryote, number of cells, and we look at number of cells here. If this is going to be unicellular, then automatically this is going to be multicellular. All right, so this was prokaryote. So if this is unicellular, this is going to be multicellular. I will move on down, and we move on to 3. Now, this kingdom Protoctis, all right? And these guys are responsible for the death of most persons who suffer from AIDS. Now, here we are going to have Protoctis. We are thinking about things like the Spirogyro. We're thinking about things, I think, like the Euglena. But the most popular one for the CXC syllabus is, of course, going to be your Amoeba. Oh, wow, I've taken away your class. All right, so the most popular one there for 3 is going to be the Amoeba, which is frequently captured on the CXC syllabus. So I'm going to give you back your class. I'm so sorry. I just got carried away just now. I'm very sorry, guys. I'm very sorry. Let me just go back to your stuff. All right, so we move on to 5. This is going to be 5. What is going to be the kingdom here? I'm so sorry, guys. What's going to be the kingdom at 5? Kingdom, an example of an organism in that kingdom is pretty much the fish. So what's the kingdom there? The kingdom, what kingdom would we find fish in? Fish would be a part of animale. All right, great. Some might say animalia, animalea, animal, or animal, but animale. All right, great. So now we're going to be looking at the other, what we want to answer. Great, guys, you've been doing wonderful. Let's look at this. Left is to right, as up is to down. So if this is autotrophic, so here we have echotrophic organism. They tend to rely on others for food. Example would have been fungi, which is of course a kingdom, and an example of fungi would have been the yeast. Now, here we have echotrophic, over here we have autotrophic, and if we scroll on down just a little bit, we want to know autotrophic. What is the preferred kingdom? What is the preferred kingdom here? What kingdom would we have here? Let me see what you're having over there. So the kingdom is Plante. Screen resolution, you see better. I am so sorry, guys. I can only use that which I have. I'm so sorry. I am trying my best to help. I can't do much. But we do accept donation here. So we'd gladly accept one of those high-tech computers that we can push this content better. So we understand the limitation, but we just have to use the resource that we have. We are not sponsored. We are not funded by anyone. We are just funded by the love of Christ. And it's the love of Christ that we're using to share with you. And we would love for you to share with us something that we can do things a little better. All right? So great. Thank you. We do understand and apologize for probably the resolution that we're pushing, but we can't do much better now. All right? So we understand and we empathize with the position. All right, great. So figure 3, one of the things we could do is to follow on the paper. I hope that we have a paper and we are filling in as we go along. All right, so we are looking now, we want to come, well, we did that just now, we got 5 marks. So each kingdom is further subdivided into taxa, or into taxa, or taxonomy, with the lowest rank being the level of species. Now, I think everyone can relate to species. And what we want to do now is to define the term species. It's always spelled with an S at the end. All right, so define the term species for me very quickly. I think everyone should be able to do that if you're a biology student. You can't be a biology student and not know what the word bio means, or photosynthesis, or species. All right, great. So let me see what species here now. Let me look at it. Species is the most exclusive classification of organism. I'm sorry, that definition doesn't help us any at all. All right, a species are a group of organisms with common ancestry and are able to interbreed. I have seen this so much time, to produce fertile offspring. That's a good definition. Or the most specific classification. They are, but that is sort of vague. One of the things we want to look at when we are looking at species, we must do the definition to state that they are able to mate and produce viable offspring. When we talk about viable, we're talking about fertile offspring. So let us look at the example here when it comes to species. We have to know them. Now, if you take, for example, I don't know where in the Caribbean you are and how much you are exposed to these stuff. If you have a horse and a donkey mating, then a possibility of the offspring is going to be a mule. But one of the things that we know about the mule, though the mule is a hybrid organism, the mule tend to have different chromosome number. Just like the horse and the donkey, they have different chromosome number. What happens is that the mule will not be able, in most cases, 99.9999 cases, the mule will not be able to have an offspring. The same is true for the offspring of a tiger and the lion, which is a liger. It is a hybrid organism, but it does not produce a viable offspring. Now, if organisms are of the same species, they are able to interbreed and produce a viable offspring. Now, a viable offspring, or a good example of organism and the use of the word species, is to look at the whole idea of these two rats, Rattus rattus, Rattus narvinchicus. Now, there we are looking at what some person call a scientific name or the botanical name if we were looking at plants, where let us look at human very quickly. Homo sapien. So Homo would have been our genus name and sapien would have been the species name. Now, any organism that would have had that species name sapien, they would have been able to produce offspring that are able to also produce offspring, carrying on the genetic trait for your family. When we look at these two rats, Rattus rattus and Rattus narvenchicus, if you observe these two rats, the only difference between them is that one is black or gray and the other is brown. While they share the same common genus name, which is Rattus rattus, if you look at the species name, they are different. So narvenchicus and Rattus, two different species name. Though they are both rats, they are not able to interbreed and produce viable offspring, all because they are of different species. So once you're going to be talking about species, in your definition, you must make known of the fact that if they are of the same species, they are going to be able to interbreed, or they are going to be able to mate and produce viable offspring. What's happening here? Organism with the same characteristic and can interbreed and produce viable offspring. Remember, I just told you that the horse and the donkey, they are not the same species, but they can interbreed. It is not that first breeding is the problem. It is the second breeding where the offspring of the horse and the donkey will not be able to produce an offspring that will be able to produce an offspring or another offspring. All right, great. Thank you very much. Let's move on. It's getting a little tricky now. And most of what we're going to be looking at now, if you want to go to evolution, which is in the back of the book, or evolution, mutation, Charles Darwin, that type of a thing, you're going to need to arm yourself with that bit of information in order for us to look at the rest of what we have to cover. We are on page 9. I'm not even been following what I have here. All right, great. So we are on page 9, and it's going to get a little trickier now. And then after this, we're going to be looking at the menstrual cycle, something I really want to do, because the prevalence of the menstrual cycle and the female reproductive system on the paper, it is so prevalent, we really want to do that, should in case we see it. So let's look at this question. The question here is saying, using the human species as an example, explain why physical appearance or morphology is not always useful for identifying organisms belonging to the same species. So why is it not always useful to just look at an organism and pretty much classify that organism or identify the organism as belonging to the same species? Why? Tell us in the chat. Share your answer with us in the chat. In order to know about the dog, first you need to identify what is the species of the dog. Now, and this question might be nice, the second question, because you might be looking at the dog, and because one of the dog is gray, and one is orange, and one is blue, and one is pink, you might think that the dog is not of the same species. But the phenotypic disposition of the dog does not dictate that the dog is not of the same species. And that is the question that we have to answer in question, I think 3. The question that is on the screen now. I don't know if you can see it. I could make it a little bigger for you. Not the look, but the genes. But why? What's your explanation? Because physical appearance is deceiving. Let's go a little. Because organisms have different physical characteristics as a result of meiosis, a medical condition which affect physical appearance. There's this man somewhere that took some wrong medication and his skin color changed to purple. Because he has a purple skin, does that mean that he's not a part of the sapien species? Environmental factors. All right, every organism. Okay, my answer is that they may look alike, but not belong to the same species. Example, if we look at these two birds, we have the bird that we refer to as the Western Meadowlark and the Eastern Meadowlark. These are two birds that look black and yellow. If you see them, you are going to be convinced that these are the same birds belonging to the same species, but they are not. So you can't just look at an organism and just say, oh, wow, they look alike. They belong to the same species. No. If you look at these two birds, the Western Meadowlark and, of course, the Eastern Meadowlark, you could Google these two birds. And when you look at these two birds, they are pretty much identical, but they are from a different species. All right? So that is pretty much my answer. Then they are not able to interbreed because they are of a different species. So the looks are pretty much deceiving. You can't look at the organism to say that they are the same because they might have different genetical makeup and, of course, not being able to interbreed. So that's pretty much my answer. We spoke about the Rattus rattus and we spoke about the Rattus narventricus, and I could go a little further. Now, this is not to do with species. But if we were supposed to look at Jamaica as one of Jamaica's biggest problem, the problem in Jamaica that they call paternity fraud. And for the street term, jacket. When a man is exposed to jackets or paternity fraud, most persons are usually the man is saying to himself that that is my child. The child look exactly like me. The child does just about everything like me. Only to do a DNA test and find out that you and the child is related so far. It is impossible for your brother, your uncle, your cousin, your nephew, your niece to be related to this child, much less you. So we can't look at something and just say, oh, yes, they are related. We are going to need to do some DNA test. Yes, jacket in Jamaica, not from Jamaica. Okay, great. Jacket, paternity fraud. What it means is that a woman would have assigned a father to her child. It is not the biological father, but the woman so decided that X is the person I was with, and I'm going to give X the child. It is one of those common things in Jamaica. I am sorry, but it is the truth. If you read the literature, it is a common thing in Jamaica. Jackets. All right. So we are we know we are sensitized to the fact that we just can't look at things and say that they are what we think they are. We need to do some biotesting. But I knew what it meant. Okay, great. You're not from Jamaica, but you knew what it meant. Okay, great. All right, let's move on. It's not a good practice, and we could talk about that more when we look at genetics. So just one reason why biologists use biological concept of species instead of physical characteristic to classify organism. So why we don't just look at an organism and classify this organism as whatever, instead we are using the species classification for that organism. Tell me in the chat what is your answer, and I'll also share my answer with you because I know that it's a little tricky here, which means that you have to go and read some more on evolution. You would have to read more about the peppered mutt. You'd have to read more about the giraffe and the long neck. You need to read more about the finch of the Galapagos Island. If you are finding difficulty answering these questions, it suggests that you need to do some more reading over there. Okay, paternity court. Okay, great. I see. This is because the use of biological concepts shows that reproduction can occur through physical characteristic, though physical characteristic may show variation. Ah, 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 ah. Interesting. How would I answer that? Because there are factors that change characteristic of an organism. All right, this is my answer. When you classify organism by species, it ensures the specificity of the organism. It ensures the specificity of the organism. Now, I'm going to give you an analogy afterward. What else do I have there? Hence, there is no problem with breathing the organism. Let me give you an example of an analogy. Might be a little far, but it should help you somewhat. When you look at species is the lowest rank of a classification. King Pharaoh called only female generals softly. So when you reach down by softly, you are being very specific. So I think somebody, let me look, Blair, I don't know where Blair is from, or Casanova, they are from some other island. And they wanted to find Mr. Wilson. Yes, Mr. Wilson lives in Jamaica. Are you going to just hop on that plane and come to Jamaica and expect to see Mr. Wilson? No. You want to find somewhere, you want to be a little more specific. So I would say that probably I live on Mullines Road. Are you going to walk the entire Mullines Road? Mullines Road is going to be like the genus. You still can't find me. But if I were supposed to tell you that I am living at 20 Mullines Road, that would be like my species identification. When you come to 20 Mullines Road, that is exactly where I would be. So when we use that species classification, we are sure, we are certain it is specific. We know what we are talking about. Like if we were supposed to talk about chordates. Chordates, all of these organisms are backbone. But are we talking about a reptile? Are we talking about a bird? Are we talking about mammals? What are we talking about? So the truth is, when we use the specificity of species, it makes identifying organism easier and more efficient. All right? Even when we are going to put them in zoos and so on, we are better able to think about how to care for them and all of that, because we understand pretty much what the species identification for that organism is. Yeah. All right, great. Let us look at this one. We are going to finish this paper, guys, so don't fall out on me now. I know this is a tough question for you. Look at this one. You would have seen this in some cartoon. Some lizards were washed away on a log during a hurricane and landed on a remote island. Several years later, they were classified as a new species. Using biological concept of species, state 2 reasons why they were reclassified. Now, I want you to pay attention to the clue in this question. Species. So if we know what is species, we already know what the answer, at least one of the answers should be. And like I said to you, all of what we're looking at now, you want to go to your textbook, look at the evolution. You want to look at things like Charles Darwin, what he said. You want to look at the finch on the Galapagos Island, which would have been a primary example for this. You want to look at the reason for the giraffe neck being so long. You want to look at the reason why the pepper must change the color. All of these, if you can't answer these questions, it means that you need to do some more reading there. It's not a lot to read, but if you go there, it would help. All right, what is your answer here? What's your answer for this question? You are watching Mr. Wilson live. We're doing our marathon session for the biology students who will be sitting the exam paper 2 on Monday. If you like what we do here, please be reminded to like, share, and of course, subscribe. All right, so we're looking at natural selection being said on the screen. It may be that they have interbreed with lizard on the island, or just, okay, we're getting a lot of things here. All right, so let me just speed it up a little bit. Now, if we talk about species, let us look at the question again. It's a we're supposed to use the biological concept of species and state two reasons why they were reclassified. Now, if I'm going to be looking at species, the first thing that is going to come to my mind is that they were not able to interbreed. And now that they are not able to interbreed, then we can't consider them to be the same species, because if they were the same species, they would have been able to interbreed. Another thing that I think is that they would have had some changes in their genotypic disposition, which would have impacted the phenotypic disposition. So though they tend to have some real difference. If we were supposed to look at the finch on the Galapagos Island, the difference in the finch was pretty much the beak, the bill. Some were thick depending on what they were eating, and some were thin. So those that were eating the harder stuff, they had a very thick bill, and those that were eating fruits and so on, they had very small bill. So my answer there would have been 1, because the organism were no longer able to interbreed, and 2, because there was some real changes in the phenotypic disposition that was pretty much influenced by genetic changes or genotypic changes. That's my answer there. Let's look at the other one. The new species of lizard living in the mountainous region of the island were found to be smaller than those living on the lowland. So just 2 reasons for this variation. Now, like I continue to say, I gave you some examples that you could turn to. Now, what is your answer for this? Some persons are saying mutation. Yeah, but mutation would have taken place, and because the mutation take place, it would have caused some changes. Either it's going to cause them now, the mutation cause them no longer to be able to interbreed, or no longer are they looking alike. So yeah, it could have been as a result of mutation, but what will the mutation really cause? All right, so we are looking at the final question here. Is it the final question? Yes, I think it's the final question for this section, and then it gets a little easier, and then we can have a smooth flow to the end. I realize that persons are tired. I'm tired too, but I am committed to helping you, so I'm here. Okay, the new species of lizard living on the mountainous region of the island were found to be smaller than those living on the lowlands. So just 2 reasons for this variation. What do you have? All right, what are the factors that are going to influence changes in an organism? Now, a lot of things. You could have environmental factors, you could have genetic factors, and you could have, as I have seen here, mutation. All of these could have impacted the changes in the organism. Now, here we're asked, let me just read it one more time. The new species of lizard living in the mountainous region of this island were found to be smaller than those living on the lowland. So there's 2 reasons for the variation. Now, 2 reasons for the variation. One, the lizard could have evolved to survive. So they have changed in order to escape their predators. So they tend to, over some time, becoming smaller, because the places that they're hiding is so small. So over time, they start becoming smaller. That is one of the things that we could look at. It could have been that there is some mutation that took place that would have caused this to happen. Another thing, so we talk about genetic factor, where over time, evolution took place and it changed. It could have been a mutation, something happened and the lizard mutated and it's now becoming smaller. Or it could be environmental factor, where there is not enough food. So after some time, the lizard is becoming a little smaller than it used to be as a result of not having any food. So the changes in an organism is always going to be dependent on 3 factors. 1, mutation. 2, genetic changes, and 3, environmental changes. All right, great. So we're going to be moving on to the other question. I realize that everybody seems to get a little quiet now. Probably need to fetch yourself some water so that we can move on with the question. Okay, great. This is the question that gives a whole lot of students a whole lot of trouble. Now I'm going to just carry the screen down a little bit. This is just question 3, you know. Are we doing the entire paper tonight? All right, so this is question 3, and we are looking by now, most person would have reckoned that we are looking at the menstrual cycle. Now we are looking at the menstrual cycle, and what we're asked to do is to identify hormone X and Y and the event occurring at Z. So we are identifying X and Y and the event occurring at Z. So quickly tell me what answer this for me. Give me that. So you are going to put X, what it is, Y, what it is, and Z, what it is. You realize that the more I am involved in this thing and you are not involved, then the more you are going to become bored. Okay, Z is ovulation. Okay, could you zoom it? Just let me zoom it for you. Great. Oh, wow, zoom. All right, so there I would have zoomed. So we want to know, this is Z, this is X, and this is Y. All right, so we're answering now. So X persons are saying that it is estrogen, OES, and persons are saying Y is progesterone, and what is Z? Anybody said what Z was? What is Z? Anybody identify Z? Anybody identify Z? Can you identify with Z? Okay, so Z is ovulation. So you got full marks for that. Beautiful. You are doing well. You got full marks for that. Great. So we can move right along. That was easy. So here we have estrogen and estrogen. Okay, I'm not going to touch more on that. Let me just go through the question. You want to look at this. Try to understand the diagram here. All right, so this is the end of menstruation, and it is pretty much the start. Here we have the start of the menstrual cycle, which should last about 28 days. Once you are seeing the start of the cycle here, we know that the hormone that comes first is going to be the estrogen. And then right as we peak, we are going to have ovulation taking place, an egg being released there. And then after that, in order to maintain here, what is going to happen is that we have to have the progesterone for the corpus luteum to continue developing and maintain should I say posture? All right, great. So we move right on. What is the role of each of the hormone in the menstrual cycle? So tell me, what is the role of the two hormones you made mention of? You made mention of estrogen and progesterone. That is not something that I need to do. That is something you can do. So tell me the role of estrogen and the role of progesterone in the menstrual cycle. Role of estrogen and the role of progesterone in the menstrual cycle. All right, so you're going to share with me the role of estrogen and the role of progesterone in the menstrual cycle. Stimulate the division of cell. Estrogen thicken. Okay, estrogen thicken or starts the development of the lining of the uterus wall. Yes, that is true. And then what progesterone does? Yes, estrogen causes the wall of the uterus to become thick, and it pretty much stimulates that rich supply of blood to that area. So what's the use of progesterone now? We're going very quickly here. Nice. I'll speed it up a little bit. All right, prepare for baby. So what it is going to do, it is going to maintain that wall. The progesterone is going to maintain that wall. That's very simply put. All right, let's move on very quickly. Now, you are supposed to name the organ that produces estrogen and the hormone that produces progesterone. What do you have? The hormone that produces estrogen, sorry, the organ that produces estrogen and the organ that produces progesterone. What do you have? This is a very easy and short question. It's not long like question number 1. Organ. All right, yes. Y maintains the lining. Yes, Casanova. All right, great. If I'm not calling the things out that you are doing, don't say much. What I do is just to grab an answer and go with it so that we don't spend the entire night. All right. Peter Gay Campbell, the pituitary gland. Oh my. No, when we do that, we are going to go in a whole big circle of the pituitary gland stimulating this and the pituitary gland stimulating that. So to make it very, very easy, estrogen is produced by the ovaries, right? And estrogen is also produced by what we refer to as adrenal gland. And the estrogen is also produced by fat tissue. So I give you 3 places where estrogen is produced. Yes, ovary, adrenal gland, and fat tissues. All right. Then the progesterone, it is pretty much produced in the gonads, and the gonads here we're talking about the testes and the ovaries, and of course they are also produced in the ovarian corpus luteum. All right, so it is also produced in what we refer to as the adrenal cortex. So a whole lot said there for why, a whole lot said. All right, so let's up on to the other question as we try to bring this down to go prepare for tomorrow. Discuss the fate of the following structure. All right, the fate, what happens to the following structure should fertilization take place? Now, we're looking at corpus luteum and we are looking at the uterus lining. Now, I wanted to leave those 2 questions as your assignment, so you can give that to us tomorrow when you log in at 5. What is the fate? So when it says fate, here, it is asking what happens to it for those persons who don't know. But I want to look at this particular question though. The contraceptive, it is like on all the papers that we have planned for you. I don't know, so we need to know about contraceptives. So some contraceptive pill containing hormones. All right, some contraceptive pill contain hormones, suggest how this may prevent pregnancy. Some contraceptive pill contain hormone, suggests how those pills that contain hormone, how is it that they prevent pregnancy? Some contraceptive pill contain hormone, how is it that they prevent pregnancy? You are watching Mr. Wilson Live Biology 2017, paper 2, and we are hoping that tomorrow morning we will have another paper. I think we have the Biology 2021, the Biology 2021 paper. I'm not too sure which other paper we'll be doing tomorrow, but hopefully tomorrow morning we will have another paper we can share with you. I think school is out and some person will be going to exam, but we're going to be having another paper we can share with you. I have 2020 here with me, and I have 2021. They are right here with me, so not too sure which one we are going to do tomorrow. So let me see what's happening here. Okay, great. It could prevent ovulation. That means that no egg will be released from the ovary. It could cause a mucus in the cervix to prevent sperm from entering or getting to the egg. Yes, all those are correct. And it could make the cervix, yes, the cervix mucus, Tomorrow morning, 5, we want to try to see if we can get there. If I'm not there, don't kill me, all right? Good, good. So we could have, of course, all those that you have made mention of, those could, of course, take place. They could, that's pretty much how the contraceptive would work. All right, so let us look at what the other question is, and I think this is going to call, all right? So some suggest, so the suggestion is here. Always don't leave a question that says suggest. Once it's plausible, work with it. Look at this one though. A drug company develops a male contraceptive pill. Suggest 3 ways in which this could prevent pregnancy. Now, if I were in the question, Audio, audio. 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 All right, great. I think we're back online. Using a different microphone, but I hope that you can hear me now. All right, great. I think we're back online. All right, great. So what I was saying earlier is that the next question after we would have looked at the reproductive system, the menstrual cycle, the other question is going to be about the eye. And I was saying when we look at, oh, it's Antindale. When we look in Antindale's summary book, any of those diagrams that you are seeing on the eyes, any of those could appear on the paper. We observe that the eye is so, it is on the paper so often there, it could be on the paper this year again. So we're trying to bring up the 2017 paper that we are looking at and to see if we can bring closure to the evening. We have about 2 other questions to look at, and then we are out of here. I do hope that you have the hard copy and you are able to treat what we are doing. It's not so difficult. I think question number 2, is it question number 2? Yeah, this part of question number 2 was a little challenging for a lot of persons. We're looking at the menstrual cycle. It's not so difficult for a lot of persons so far. We did say that we're going to leave corpus luteum and the uterus lining for tomorrow. And we had asked you, this is where we left off, a drug company is developing a male contraceptive, suggests 3 ways in which this could prevent pregnancy. I was about to say, if I were in your position, the same ways that are the same way I would have given here as to how a contraceptive pill work, I would pretty much say pretty much it would work the same way here. So for me, this is like a ABC. If you are able to answer one, you'll be able to answer the other. But let me hear you say, tell me in the chat there how it is that that contraceptive would have worked for the man. In your opinion, 3 ways in which a contraceptive could work for the man. 3 ways your contraceptive would have worked. All right, so 3 ways in which your contraceptive would have worked. Just tell me. So we're looking at the final question. I don't know if it's a final question, but we're looking at this question. Is it the final? Yeah, it's a final question here for this. All right, so we want to find out 3 ways if you are developing a contraceptive or the company is developing a contraceptive. We want to know about 3 ways in which you think it's so. 1, good, we are here now. All right, so 1, the contraceptive could kill the sperms. It could also cause no sperms to be produced. I like those 2 answers. It could prevent the male from producing sperm cell. Yes, but we have 2 so far. We need another. So 1, it could kill the sperm. 1, it could prevent the sperm from producing. What else could happen? Okay, so it could reduce the production of testosterone, prevention of sperm, production of sperm. Yeah, it could prevent the sperm from being produced. It could also block the vas difference. That just like when you what they call, what is it, tubal ligation? Is it who does tubal ligation and who does vasectomy? The male does tubal ligation or the female does tubal ligation? So those are two methods of contraceptive. So it is pretty much like putting a barrier in the tube. The hormonal pill could do that to prevent sperm from moving through that tube. So pretty much that was very easy for a lot of persons. All right, the female does what? Or the male does vasectomy and the woman does tubal ligation. And if you can't remember, just remember women are always saying that they have blocked tube. Right? So it's almost fair to say tubal ligation is for the woman while vasectomy is for the male. All right, great. So the question that we have here, it's question number 4. It wants us to explain. I'm reading from, I'm like overly prepared now. I have paper and I have a computer that anything fail I can work. So explain how the structure of a eye control the amount of light that enters the eye. Do that one very quickly. That is not so difficult. Explain how the structure, remember we spoke about structure when we were talking about xylem and phloem. Hello, good evening and welcome to the show. Remember to give it some like. All right, how does the structure of the eye, how does it control the amount of light that is entering the eye? The structure of the eye, how does it control the light intensity? Okay, great. When I start to see the radial and the circular muscle, I start getting excited. Radial and circular muscle contract and relax. Hello, Israel, you can't put that on the paper because I don't know which one contract, I don't know which one relax. So you want to be a little more specific, all right? Okay, so the CXC topic so far were right. They didn't trick us, it seems. The circular muscle contracts, and when it contracts, what happens? Yes, the circular muscle contract, but when it contracts, what really happens? All right, so I am waiting for the answer there. So in dim light, the circular muscles will relax and the pupil will, of course, become widened, while the radial muscle will contract. All right, so when the circular muscle relaxes, then the radial muscle will contract. Now, the opposite is true for bright light. So one of the things we need to remember is that once we're in dim light, the pupils are going to become very, very large to allow a lot of light to pass into the eye. And this is as a result of the circular muscle being relaxed and the radial muscle contracting, opening up the pupil, allowing more light to go in. And the reverse is true if you are in bright light. All right, great. While we move on to the other question, I'm just going to share something with you very quickly. All right, so I'm just going to put something on the screen very quickly. I hope you can see it. All right, so that's pretty much an extract from my book that comes out in August. Just a little bit of ads here, and then we are going to up right into what's nope? Somebody said nope. What happened? Nope. Do we have an I for paper 2? When you subscribe to the channel and you click the notification bell, you get to be, you have to have a YouTube account in order to do that, right? And you are informed whenever we are having anything, when we are sharing any content, you'll be informed. All right, so we can just go back to the paper now, HSB and bio. All right, so that's the extract from the book. I'll show you again sometime during the session. All right, great. So we are ready to continue. So next question, from question number 4, we move on down and look at this question. This here is looking at the retina. Now Jenny enters a dimly lit room and cannot see the color of the furniture until the lights are switched on. Now explain how Jenny is able to discern objects in the dimly lit room but cannot see the color until the light is turned on. So I'm going to just magnify that you can answer, because a lot is to be said here. So just let me just ensure that you're seeing it well, and you are going to explain this for me, what is happening here. So we are in the retina now, and by this, once we are seeing light and dark and the retina, we start thinking about the rods and the cone. The eye. Okay, so remind me, the eye is not there? I remember that respiration is not there. The eye is not a part of the broad topic. Just say no so we can just skip on. The eye is not there? All right, I really don't remember. So if it is not there, and I like to have a guardian, so if you say no, we are not going to be doing that, just like how we have passed the respiration on most of the paper. Let us just do this then. Once you say it's not there, I do believe you. I sent out the broad topic. I can't tell when. Oh, it's not there. All right, great. We're not going to do it. All right, what I'm going to do, everybody can do the genetic diagrams. Yes, no? Do I need to do this? The genetic diagram? Do we need to do the genetic diagram? Yes, no? It's your class, you know, so tell me what I need to do. Okay, great. So, okay, all right, great. So, being that you say that, I'm going over there. I'm going to increase my screen somewhat. Remember, I am using my touchpad on the laptop to write, so don't kill me if things are not written well. I just want you to follow and write in your book what I'm going to say. Now, look at this. A certain form of color blindness is inherited as a recessive allele. Important that we look at this. If you don't know that color blindness is recessive, now you know. And it's carried on the X chromosome here. It is said to be an X-link or sex-link. Now remember that we have Y-link, but we're not looking at Y-link at CXC, we're looking at X-link. A woman with a normal color vision, whose father, look at the distraction here, look at the distraction here, whose father is colorblind, mates with a woman, the woman father was colorblind. That's her father. Now, she mates with a colorblind man. What is the chance of her having colorblind children? Use a genetic diagram to explain your answer. Now, usually, when we are doing this, the first thing we want to do, we want to define the alleles. Usually we start with let. Now, if you observe here, it is already defined for you. Now, the fact that the woman father was colorblind, this is a sex link. So if it is sex link, remember I said this is going to be X on that chromosome, and the Y is a little shorter. So because it's this short, to put it in a very simple way, it doesn't really accommodate that genetic trait that we're looking at. All right, so it would be something like this. I'm going to rub this out. So that is for the male, and if we're looking at the female, then we would have XX here that represents a female, and I am trying to explain what is happening with the father. Now, because it is an X-link trait, what you're going to find happening is that for the father to have it, the only thing you need to have is the trait on the X. Now, while we talk about the trait on the X, let us be reminded that it is the man that determines the sex of a child. See it? This is what represents a woman. XX represents a woman, and XY represent the man. Why the man determine the sex of the child? If the man was supposed to donate this over here, then the child would be born with XX, making the sex of this child a female because the child has XX chromosome there. Now, if the father was supposed to give the Y, then you understand that it would have been XY, and of course, it would have been a boy. That being said, let us just erase this and let us get to the matter at hand. Now, if the man can only carry the allele that we're looking at on the X chromosome, this is going to be the man. So this is our father, X, and he has the recessive allele there being B. Now, we know that it is because of the father why she's a girl, because he gave this X. So naturally, we also know that this girl must have had the trait for color blindness. So we know naturally that this woman is a carrier because she would have gotten this from her father. Let's erase that. So just by the fact that we know what the father is, we can say what the daughter is going to be. The other thing we know is that this woman had a relationship with a man that was colorblind. Now, if the man is colorblind, like her father, he would have something like this. So this is the man that she married. All right? She married to that man. And this is her. Great. Now let us look at how we solve this problem in the exam. There are some things that we should do and some things that we should not. So let us start out. The first thing that we want to do is to identify the parental genotype. Sorry, the parental phenotype. So write for me. I am not going to spell out everything. I can't write this. So I want to talk about the parental phenotype. So the parental phenotype here for the offspring between this woman and this man is that the woman is a carrier, or some might say that she's a normal carrier, and the man is, of course, a colorblind individual. That is the phenotype. So he's colorblind. You can write out that. So that is the parental phenotype. That's the first thing you need to write. Now, usually we would have defined the alleles here. We would have defined the alleles here. And then the next thing, so 1 would have been to define the allele. 2, to write the parental phenotype. 3, now to write the parental genotype. So we're going to have PG for parental genotype. Now the parental genotype would have been here for the woman. It would have been, being that she's a carrier, she's going to have a capital B because the B is defined as being normal here. So the B there defined. And then she's going to have that recessive allele, which is of course going to be this common B. So this is mother. Now the genotype for father is going to be, he's a male, so it's going to be XY because he's a male. And he has the disease, so he only needs to inherit the trait on the X. Remember, the trait is not on the Y, it is always on the X because we are doing X-linked inheritance or sex-linked inheritance. 3, the next step we want to do now, the 4th step, is that you are supposed to write out the gamete. To write the gamete, all that we do, we are going to write XB over here, XB there, and then over here, we're also going to write this XB. If you observe, all that we did now was just to space them. This line here is just to show, it's an arrow to show you what we did. So that this is now the gametes. So gametes, please write it out as I say it. I can't write it my touchpad, the gamete. And then here we're going to do the same thing. So we have little b here. Oh, wow. Anybody see that arrow? Okay, this is supposed to be a common B. Let me erase that. Sorry, that is supposed to be a common B. So there we have a common B. Please pay attention, guys. All right. And then all we do now is to carry down the Y over here. All right, so we can all erase this part here. So these are some things that we need to do before we start doing the genetic diagram. Now, the truth for me, I like to tell my student all of those line crosses doesn't really make much of a sense. Because when you get to college and you are doing like dihybrid cross and trihybrid cross, you really can't draw that amount of line to solve it. You are going to be confused. So the 5th stage is to do what we call random fertilization or segregation. All right? So let us start again how we solve a genetic question like this. First thing, you are going to define the allele. Second thing, you are going to write the parental phenotype. That is what the parents look like, that type of a thing, as it relates to the disease you are looking at or the trait you are looking at. Then the next thing, we are going to write the genotype for the trait, for the parent. Then we are going to write out the gamete. You must follow this particular order. Then we are going to do segregation. Now, segregation or random fertilization is the same thing that you call it, you draw the line. But I don't like to teach students about line because by you finish drawing the line, you are confused. So what we do, we draw a Punnett square. So here's our Punnett square. All of the Punnett square is a rectangle with 2 vertical lines. I can't, if you're writing in the chat, I can't see you right now here. So when I come back over, I'll look over there to see what I need to look at. So we have 2 vertical lines. Then we have 2 horizontal lines. And remember, I'm not using a ruler here, so just bear with me. So what we're going to do, some persons can't remember male from female, but in this case, you need to know the male different from the female. So here, we're just going to write the male on top here. And we're going to write the female right here. You could use a symbol or you could write the words out. So we're going to put the male on top right there. We're going to put in these here. So we're going to have X with a B, and we're going to have the Y on this side. Then these now, we're going to put these over here. We have the capital X there with the capital B, and then we are going to have a common X, sorry, a capital X with a common B. Now, what we're going to do, wherever these intercept, we are going to write both in the cell. So we have capital B coming from over here, capital B, and we carry over the X with the capital B, and we have another common B there. So here, the first organism, We have the genotype for the first organism. You need to also know the phenotype. Remember, the phenotype is that which you can see. And I like to say that the genotype is these letter stuff that we are using. It's an easy way for students to remember. All right, so there we have it. We are seeing both male and female so far. And then now we're going to look at this individual. This X, it comes from here, and it will put a little thing there. And this X here, it comes from here. And we put this here. So you see this, they intercept here, and we'll put them both in the cell. Look at this here. And we are putting a common B there. And the Y doesn't have anything, we'll bring down the Y. So if you look at what we're having here, we could look at the chance. Usually the question is about the chance. So let us look at what it says here. How did I get here? Okay, it says, use genetic diagram to explain your answer. It says, what is the chance of them having a child that is colorblind? Now, what I'm going to do for you to bring you back into the class, I'm going to number these now. So I'm going to use a different color, and I'm going to number them with green. So this is going to be 1, this is going to be 2, this is going to be 3, this is going to be 4. I want you to identify the phenotype for 1. So tell me in the chat, what is the phenotype for 1? What's the phenotype for 1? What's the phenotype for 1? Tell me in the chat. So what we have on the screen there is all genotype. Now we want to find the phenotype. Okay, so 1 is normal vision. But hold on a bit. That would pretty much refer to a phenotype, but you can't, while the person is normal, has normal vision, the person, it's not that's not the way you're going to write it. First, you're going to write the sex because this is sex link. So you're going to write female because it's XX, so it's a female. So it's a normal carrier female, or a female carrier normal for 1. What that means is that this is a female with the trait. And with a trait, she's not affected by color blindness, but she's carrying the trait, right? So that once you talk about it, she's not normal. She's a normal carrier. Normal carrier. A normal female would have had 2 capital B's, right? So she's a normal carrier. She's not affected by the disease, but she's carrying the disease around with her. Now, what is the phenotype for 2? Okay, so 4. Yes, 4 is, you can't just say blind. You have to say the sex. You have to say the sex with the trait. So you have to say it's a female colorblind, and the person is not blind. It's that the person is not able to see some color. So 3 is a female colorblind individual. All right, so we are trying to identify each. So just let me tell you them now. So 1 is a female normal carrier. That's how it should be written. 2 is a normal male. 3 is a colorblind female. That's a phenotype. And 4 is a colorblind male. What is the chance? That is what we're supposed to answer. The chance here is that there's going to be a 50% chance that from this interaction, you are going to have a child that is colorblind, with 25% being girl and 25% being boy colorblind. That is your chance. All right? So number 4 is going to be a male that is colorblind. So the chances is what we're going to offer. It's at 50%. You can't leave it at 50%. You can't just leave it at 50%. The chance is that 50% will be colorblind. We want to know, and if you are using ratio and you are saying 1 and 1, you must also say what the 1 means. All right? All right, are we all fine? So I'm going to be moving on. I have a video on my channel that covers all of these stuff. I would have done these some time ago. You can just go there and check the genetic playlist and you'll have these all worked out. Now, this is a very long question and I'm not going to do it on the show, but I'll just give you some pointers as to how to answer this question. It says, describe the chemical digestion of carbohydrate, fat, and protein from the mouth until it gets to the absorption in the small intestine. Now, we would have known that carbohydrate starts to digest in the mouth, and the mouth pretty much has a pH of about 7, pretty much alkaline thereabout. And of course, salivary amylase is acting on it. In answering this question, you need, because it's chemical digestion, you need to say the name of the enzyme, the substrate that it is acting on, and the final product. You need to also say where it is taking place. Is it taking place in the mouth? Is it taking place in the stomach? Is it taking place in the small intestine? Now, if you look, for example, at protein that has pepsin and renin in the stomach, and it has trypsin in the small intestine, you need to make note of that. But you must also take into consideration that this particular question spoke about absorption in the small intestine, which means that you are going to need to tell us about how the absorption takes place in the villus. We are telling about the fat and the lacteal, that type of a thing. You must also make mention of the bile, which emulsifies fat. Bile is not an enzyme, so bile will be breaking down fat into fatty acid and glycerol. Now, this is a very long question, so we would not be able to type up all this for you. See, this is a very long question and it's worth 6 marks. Now, we are also supposed to describe how the body system work together to assimilate glucose. So I'm coming back there to see what's with you. It falls, digestion is under nutrition like photosynthesis. Digestion falls under nutrition, pretty much like photosynthesis, pretty much like decomposer, pretty much like autotrophic, saprophytic, and echotrophic. All right, are we good? So the digestion question is pretty easy. I am not going to be going through all this. I'll just tell you where to find the answer. So if you go to your textbook here, it speaks to assimilation. So we are now just talking about how glucose is pretty much converted into glycogen and stored in the muscle and liver cell. Pretty much you could look at that in your nutrition. Just go to your textbook. This is straight from the textbook. Explain assimilation of glucose in non-diabetic person. Now, when we look at this, we are going to talk about the negative feedback mechanism. And in the negative feedback mechanism, just let me just come on over and just talk to you about that a little bit. There are two, we'll talk about homeostasis. We're trying to talk about the maintenance of a constant internal environment. Now, when we look at the control of glucose, once we talk about diabetes, sweating, eating, that type of thing, we're talking about a negative feedback mechanism. With a negative feedback mechanism, what the body does is work to bring back a state of normalcy, or normality, or normal. When we talk about the positive feedback mechanism, what the body is doing is to create more of something for a particular process, as is the case when a woman is giving birth and the estrogen is produced in order for more contraction to get the baby to move through the birth canal. Now, when we talk about the negative feedback mechanism, as it were with glucose, it's very, very easy for glucose. When you are hungry, what is going to happen is that the body will pretty much cause glucagon to convert glycogen, which is stored in the muscle and liver cell. It will convert that stored glycogen into glucose to make the energy available. However, after you've just had a diet or have your breakfast, your food, whatever, there's going to be a high concentration of glucose in the blood. And with this high concentration of glucose in the blood, then the pancreas is going to pretty much secrete insulin. Now, the insulin will convert, providing that you're an healthy individual, it will convert the excess glucose into glycogen. And this is, of course, in the aid of the liver, and it is stored in the liver and muscle cell for when it is needed. So that's the start of the negative feedback mechanism. Now, in a person who is sick, it is going to be different. The person who has diabetes, this person is having problem producing insulin, or the body is not responding to the insulin that is produced. When that happens, we're going to be having a whole lot of blood, sugar in the blood. We're going to be having sugar in the urine. The person is going to pass a lot of urine. And of course, if this is not rectified, then naturally other organs are going to start failing, right? So we want to ensure that we are looking at our simple sugar, looking at our complex sugar, because with simple sugar, it gets into the blood readily and could cause serious problem. So we want to ensure that we are consuming more complex sugar than simple sugar. And we're talking about the syrup and the donut and those sweet stuff that we eat. We might be eating ourselves to the grave. Now let's move on down. We are almost finished. So we're at question number 6. Question number 6 is going to be the easiest thing in the world. So just write that for me very quickly. Using suitable example of each, distinguish between predator-prey relationship and parasitism. I have a video on my channel on that. I'm sure that you should be able to master that. Then we come on down here. I think I need to talk about this question because this is not so straightforward to most persons. We are looking at biological control. Now, some environmentalists encourage the practice of the introduction of predators to an ecosystem to control the population of pests rather than to use agricultural chemical. Now, we are supposed to outline 3 advantages and 3 disadvantages of using the biological control. Now, a biological control is pretty much using one organism to control another. So once we're talking about biological control, we're using one organism to control another. Now, this can be a lovely practice and it can be a dangerous practice. Now, I could tell you about the dangerous practice as in the mongoose in Jamaica. That was a biological control. It was brought in to control snakes in the cane field. Having had a lot of snake and the food is not as good as it used to be, it is now eating chickens and eating eggs of birds, driving some birds into the extinction vortex. It is thought that the mongoose ate the iguana eggs in Elsha, and the iguana almost went into the extinction vortex just the same. Now, one of the bad thing about, I don't want to talk about bad first because I have a whole lot of example there. One of the bad thing about biological control, and the first thing that comes to mind is that biological control being bad, the organism could become an invasive species. Now, you would have heard about species that are endemic, meaning that they belong to the area that they are found, like the coney in Jamaica and the giant swallowtail butterfly. Those organisms are endemic to the island, and of course, they will be found here. But we have things that are pretty much invasive. An invasive species is a species that is introduced to an ecosystem and it has no natural predator. So we have, like in the Kingston Harbor, we have the Pernavidas oysters, we have the Australian crayfish in Jamaica, we have the lionfish in the Caribbean Sea. Those are invasive species. But let us hop on over to Brazil, where we brought in the Brazilian cane toad as a biological control, and the Brazilian cane toad did so nice with the beetle, and it had no natural predator. So in order to control the Brazilian cane toad, what happened is that they started to kill themselves. So the competition was really, really high, and because the competition was high among them, they started killing each other, and pretty much that is how that ended. So now it is an invasive species. They're having difficulty controlling them. Now, one of the big thing about a big disadvantage about a biological control is that a whole lot of study must be done in order to introduce the predator to the area. You must do a study on the prey. You must do a study on the predator, and this can be a very expensive setup. So that's a disadvantage. We said that the predator could become invasive, where it starts to attack other organisms, as in the case with the mongoose and the Brazilian cane toad. Another thing that stands out for me is that most one of the thing is that most of these organisms, the predator, they have to be spread over a wide area to control the particular organism. You can't just introduce it to your farm. You'd have to spread it to a large area. So that is a disadvantage, right? Because remember that in some area, the number of organisms might not be as big as it should be. And if you look at a food web, once we put in a biological control, it's like we are upsetting the food web. If the organism eat too much of the prey, then we are going to have some crash in the ecosystem. Now, another disadvantage is that the predator cannot eat all the prey, because if it eats all the prey, eventually it is killing itself because it won't have any food to eat, right? Another thing we want to look at is the advantages. The advantages is that it's going to be a cheaper method over a long-term period. And another thing is that it doesn't allow for chemical drift, as we were talking about chemical. So it will only kill the intended organism. So it is sort of specific if it is used correctly. Another advantage is that it provides a long-term solution to the particular problem that we want to address. The last question on this paper for tonight, our last question is going to be, and then I'll allow you to chat. Our last question is going to be, explain how biological control may contribute to biological evolution through natural selection. And when we talk about this, the first thing that comes to mind is co-evolution. So while biological control is taking place, an organism is evolving to stay alive. So the organism might come up with new tactics. The organism might be able to camouflage. The organism might come up with cryptic coloration. The organism might come up with speed. And while they are doing this, the predator is coming up with ideas in which they can lure the predator, they can trick the predator, they can run down the predator. They are coming larger, they are becoming smaller, and they come with a whole lot of evolutionary tactics in order to stay alive. Once you put a biological species, a biological control, we are going to have co-evolution taking place where the predator is trying to get the prey and the prey is trying to escape, and over time they are changing. So through co-evolution, the prey will develop or evolve to stay alive, while of course the predator is doing just the same thing. Yes, they could have poison, venom, and so on. We could think about things like the sea anemone and the clownfish. That's a good example there. All right, so that brings us to the end of the 2017 paper. We're going to try our best. It's 9 o'clock now to see what we can do. If we don't have this live, we're going to be having some videos for tomorrow. Now, that is pretty much my offering for this evening. I am very happy that you took time out to stay with us. Like I said, we are trying our best to put in 6 paper 2 by now on Sunday. The 6 paper 2 that we're doing, you would have already have 2016 on the channel. So we have done 2016. We just went through 2017. We are going to be doing 2018. We are going to be doing 2019. We are going to be doing 2020 and 2021. Now, 2021, I don't know how many persons would have seen 2021. Just let me pop it on the screen for you just before you go. So if you don't have it, you could get it. We might do 2021 tomorrow. I am not too certain. Let me see what paper this is. Oh, this is a 2020. (2017) 2018. 2019, 2021. I want to look at something very quickly on this paper. You see, this paper, it came with a graph. The graph is not so bad because we have done graph like this before. See, this paper looked pretty much like the 2016 paper. I want to point out something, guys. Let me tell you what I'm looking for. I want to point out that on this paper, COVID was on the paper. Was this the paper or was it the HSB paper? All right, so this is a 2021, January 2021 paper. You would have seen this diagram in your textbook when you are looking at photosynthesis. All right, so this is what we have here. The 2021, not looking so bad, but I think the 2020 paper is much easier. It's easier than them all, the 2020 paper. All right, so this brings us to the end of the evening. This brings us to the end of the evening. All right, so 5 a.m. and then 5 p.m. tomorrow. No, I don't think we're going to have a 5 p.m. show on Saturday. You might get a video, but we're not going to be having an evening show for Saturday, but we're going to be having an evening show for Sunday. I doubt we're going to have a show for tomorrow evening, but tomorrow morning we will have one out. And if we don't have a show, we'll try to send out another video. All right, that's the best that we can do. I'm still working on my book. Might I just share my book with you before you leave so that when you see it on shelves, you can tell me what you think about the back of the book. We're showing you the book back, where it is. All right, so this is just one of my books. So I have this book working on, and I put it aside to assist you with your SBA. So that's a story. Tell me what you think. You can read and tell me what you think there. That's a story. Sure, can we do this paper two, please? Which paper two? We're only doing paper two. Which paper two are you talking about? All right, the replay will be there. No, not everyone can join. If you can't join, you can catch a replay. You have to understand that I would have other things to do, so I am just trying to make it work. So if I'm not on at 5, you'll see some videos. The videos are going to run all night for those persons who need to see some videos. When I leave here now, I'm going to be running the videos all night. All right, great. So have yourselves a wonderful evening. I am going to see if I can transform into something new now. Remember to like, share, and subscribe. And when you subscribe, click that notification bell and select all. You subscribing, you watching our videos will tell us that you need what we have to offer, and then we'll have no option than to continue preparing for you. But you need to subscribe. You need to subscribe. You need to watch many videos over there. You need to call your friends on over. You need to make it happen. All right, it's a handshake. You work your side, we work our side. We're not going to be putting out videos unless persons are watching them. We're not going to be having a live unless persons are coming to the live. So that type of a thing. So we are extending ourselves to all of you from across the Caribbean. I don't know you, but I wish for you all the best in your exams, and we are doing the best that we can do. I'm trying to move over to my videos, but it's going a little slow. Oh, that's the problem. So for persons who are interested in seeing videos, I will just post a link. If you can't access them, I'll just post a link and you can go and watch whichever video you so desire. But we only do content for CXC, so you can check out what we have here. And
looking at the May-June 2016 biology paper. It is, of course, the paper 2 paper, which is slated to last 2.5 hours if you were supposed to do it, but we're going to see how quickly we can get it done. Please ensure that when you go to the exam, you read all the instructions on the paper so that you'll be answering what is asked and not what you think was asked or should have been asked. Now, question number 1 is usually a lab question, so it's very important that your lab skills are up to par. Let us read this question. It comes from section A of your paper. It says, answer all questions in this section. Write your answers in the space provided in this booklet. 1A. So this is section A of question number 1. It says students investigate the effect of temperature on the rate of reaction of the enzyme catalase. Now here you'll realize that in the first sentence you can find the aim for this experiment. They use crushed potato as a source of enzyme and place 5 milligrams into each of 7 test tubes. Here you'll realize that, of course, potato has protein if it contains enzymes. They added 3 centimeter cube of 1% hydrogen peroxide, that is H2O2, to each test tube and place them into water bath, kept at the following temperature, and the temperature are as outlined: 0 degrees Celsius, 10 degrees Celsius, 20 degrees Celsius, 30 degrees Celsius, 40, 60 degrees Celsius, and of course 50 degrees Celsius. They count the number of bubbles released per minute from each test tube and recorded the data in a table. Now this is a very interesting table. Let us look at what a table entails. Here you'll observe it has a title at the top, and it has header column. So here we have the header column, header column, header column, and this would be considered the header row. The first thing here, header row, that type of a thing. And this is pretty much a convention for a table. If you observe, the table has 4 sides and the numbers are all written in straight line coming down, and there's no missing cell in the table or no missing data in the table. If you observe, the unit is not repeated in the cell here, so we have degrees Celsius and it's not repeated here, and we have oxygen bubbles, and we really don't repeat the units in the cell, we just place the numbers there. Now we're looking at the table, and the convention for table is that the independent variable, that's a variable that the experimenter will manipulate, is going to be in the leftmost column. Now, if we were supposed to use the leftmost column here, which is test tube, we would have to have a legend for the graph. That would be a little more work. So I want to go on down to look at what we would have done. Here I would have just drawn a sketch of what the graph is supposed to be based on what I'm seeing here with the table. You're seeing that we're going up 0 up to 30, 40, 50, 60, but not that there. We're looking at what happens on the frequency. So we go here and we observe that we went up to about 15 there, so up about to 15, and then we had a crash. So let us look at what the question is saying. Great. So we're supposed to plot a graph on the grid provided on page 5 to show the relationship between the number of bubbles and, of course, temperature. So here we're seeing 2 things. It tells us what we're supposed to plot. We're supposed to plot number of bubbles versus temperature. Here it is very important that we understand the difference between the independent and the dependent variable, because it clearly says bubbles and temperature. So some students would put bubbles on the y-axis or on the x-axis and not really sure what they should do. But please be reminded, usually it is the leftmost that goes on the x-axis. So here it would have been temperature. Now, if you observe here, we are manipulating temperature because we have different temperature, and the experimenter actually decided what the temperature would have been. On the y-axis, this is not up to the experimenter. This will change as a result of the experimenter changing what is on the x-axis. So the bubbles are going to be on your y-axis, and of course temperature on the x-axis. That's the first thing that we need to know. And for that you would have gotten 4 marks. But here we're supposed to get this information from the graph. We're supposed to determine the optimum temperature for the enzyme. Now, the optimum temperature is, of course, going to be the ice point at which the enzyme works and is efficient. That would have been right here at the top. And if we want to find that, all we have to do is to look for the highest number of bubbles, and that would have been 15. So it's 15, and the temperature here would have been 40. This is, of course, the optimum, the ice efficient point, and that's right there at the top of that graph I've just drawn to explain before we opt. So 40 there we have as the answer, 40 degrees Celsius to be specific. The unit is very important. Now, working with graph is a lesson by itself, and students tend to shy away from graph because I don't know if enough work is not done there, but they're still shying away from graph. So the first thing we had to do, we had to work out the axis for this graph. But we did some things that probably would have been easier to be done before the axis. One of the things that we did, we drew the graph. So if you observe this red line coming down, I'll just show that to you. This red line coming down, showing that we drew the graph, and you're seeing the directional line, meaning that it is increasing in this way. So we drew the graph rather than just populating the grid. You're seeing the red line going all around, showing the x and, of course, y axis. And then you'll see the axis being the name. Temperature here in degrees Celsius, it is measured. And on this side, you're seeing that it is measured in the number of bubbles per minute. And then we had to represent this on the graph paper. So what we had to do, we had to work a scale out. So here we're saying each of these grid represent a centimeter. So 1 grid, 2 grid. So we're saying here 2 centimeter represents 10 degrees Celsius. And you're going to see that in the scale. Here we're saying that 2 centimeter represents, this probably is represented differently than it is here. We're saying that 1 centimeter represents 5 degrees Celsius, and on the x-axis and on the y-axis 1 centimeter would of course represent 1 bubble, and that you can pick that information up on the table. Now we get into drawing the curve. Now all lines on a graph, they are referred to as curve. Now if you're using a dot to plot the points, then you want to ensure that the dot, they are all circled. So I'm using dots here, so I'm circling just to identify the point that I am supposed to swing the curve through. So you'll observe here that the dot is all there. I'm using Microsoft Word to draw this graph, so it might not be as if you were drawing with your pencil. So the line is all swung pretty much through all these points, and it comes up at the top here. If you observe, there's not a sharp decline, there's a little curve here coming down, and that's pretty much, you should always have this little curve. It didn't just happen that boop went up like a ball and returned down. It had to sort of slow down and then came right back down to the bottom. So this pretty much represents what your graph should be, and I'm just going to just let that zoom in a little bit so you can have a better appreciation of what the entire graph should look like. Just let me show you the full graph here before we move on. So I'm going to put everything on one page to show you what a graph should look like if you are doing the graph. So that's pretty much what your graph should look like for your exam. You want to ensure. Now, another thing that a graph should have is a title, and you observe the title is given here, so there's no need to go recreating the title because it's already given. So all graph must have a title, and the title is given here, so there's no need to go recreating the title. I do hope that you have that and you are able to do your graph question should you see one for your exam. So we move on to the other questions, and students tend to say, I would want to see the question and not the answer. So we're going to try as much as possible to see how best we can do that. So explain the shape of a graph at each of the following temperature range. So we want to explain the shape of the graph. In explaining the shape of the graph, two things might be captured. Why is the graph shaped like it is shaped? And what does the shape actually means? So we want to look at, in explaining the shape of a graph, we want to know why is it shaped like that and what this shape means. These two things we need to take in consideration. So let us look at what the answer is for this particular question. So we're looking at that, 0 to 10 degrees Celsius. There was a gentle increase in enzyme activity, which is pretty much a common thing when we are looking at curves like the sigmoid curve. It starts out very, very slow and then picks up. So there was a gentle increase in enzyme activity, resulting in a gentle increase in curve of the graph, or in the curve of a graph, which showed, of course, an upward movement. So we're looking at what happened there, which showed an upward movement from 0 to 1 bubble. So there wasn't any bubble, then we had 1 bubble at about 0 to 10 degrees Celsius. That's how we explain the shape of a graph. We're looking at what is happening and what would have caused that to happen. Let's move on down to the other question. Between 11 and 40, so remember 11 is just 1 block away from 10. Remember the graph we were looking at. There was a sharp increase in enzyme activity. And let's up on right back to the graph to look at that sharp increase. So here we're looking at 0 to 10. So that's just starting off. But then when we look there, we're seeing something more like an exponential growth. The sharp increase, diagonal line going up. So we said there, for 11 to 10, there was a sharp increase in enzyme activity. And of course, that resulted in a movement up to 40 degrees Celsius, which was, of course, the optimum temperature. Now, this is reflected in the sharp increase in the number of bubbles from little over 1 bubble to 15, and the sharp upward movement of the curve. So you're seeing that the enzyme there had increased, a sharp increase in activity, and wow, we moved from 1 right up to 15. Now, what is it saying here for 40? So this is when it got to the optimum point at the top there. If I can just draw that for you very quickly. When it gets to the top there, what would have happened? So this is going to be the graph here. So when it got to the top, we want to find out what happened there. So look at what happened. It went up pretty much like that, and then it came down pretty much like that. So we want to know what happened at 40, and that is going to be somewhere there. All right, great. So above 40, and this would have been 40 right at the base here. So above 40, what happened? We want to know what happened. So here we were increasing, but now we are decreasing pretty much at the same rate that we increase. So enzyme activity, having reached optimum at the high point of the graph here, took a sharp decline, sharp decline here, as temperature was no longer favorable for the reaction. We'll understand that enzymes tend to work at a narrow temperature, and if it goes above or below that that temperature range that the enzyme works, what is going to happen is that the enzyme is going to be denatured. Please be reminded that enzymes are not organisms, so they cannot be killed. Now this created a non-functional active site to the substrate. That's a substrate to enzyme. Some connection wasn't taking place there. The reaction just could not continue. This is observed in the sharp decline in the curve to 0. So there was a real decline in enzyme activity as a result of the enzymes not being matching favorably with the active site, as the temperature would have denatured these enzymes. Another thing we needed to look at, we were supposed to write a conclusion that the students could draw from the result. Now this is going to be tricky because you want to pay attention as to what is asked by the experiment. So we are going to roll right back up to the aim of the experiment. We need to know that in order to write a conclusion. So the aim of the experiment here is to determine the effect of temperature on the rate of reaction of the enzyme. So we are looking at the rate, how the varying temperature affected the rate of enzyme activity. We are not looking at the temperature on enzymes. That's not what we're looking at. We are looking at the temperature on the rate of reaction. Hence, the conclusion should be fitting to the rate of reaction and not just to enzyme activity. So what did we write for this? We had something a little long. One of the things we observed from the experiment is that the enzyme work best at a particular temperature. We also observed that as the temperature increased, enzyme activity increased to a point of optimum, after which the enzyme activity stopped with the added increase in temperature. So the conclusion that can be drawn is that enzyme work best pretty much at a particular temperature, and if we increase temperature on enzyme as it were here, the activity will increase, but it will get to a point that it will eventually stop as the enzymes are denatured. So pay attention to your conclusion there. We're moving on to the other question, and I did promise that I'm just going to try to show you the question only. So just one reason why living organisms need to maintain a constant internal temperature. Well, all organisms, or most organisms, would have a constant internal temperature for most part. And we're looking at homothermic organisms here. We're not looking at the poikilotherms, which regulate their temperature using external means. Homotherms, they regulate their temperature from within. Now, a lot could be said here. To argue this point, a whole lot could be said. But this is what we said. To ensure that the body functions properly, as enzymes work best at a narrow temperature range. And if the temperature is off, it will affect the functioning of the enzyme. So you understand that the body is pretty much driven by enzyme activity, as reactions are always taking place. Now, if these reactions fail to take place, then you will become sick. You are not able to do what you are supposed to do. And you as a person will become unreliable because you're supposed to turn up this morning. You can't turn up because the enzyme activities are not functioning. What the enzymes should do, it's not happening. The body should do this, the body should do that. It's just not happening. You have this temperature up and down, and it's affecting the enzyme reaction as is seen on the graph. You want to keep it pretty much at that range where things are constant, and of course you can be reliable to yourself and of course to others. What is the other question asking for? So it wants us to look at, we want to describe 2 precautions that the student should take when doing this experiment. Now, precaution seems to be a troubled word in science. Precaution here is looking at things that you can do or should do, or things, it looks at things that you can do or should be done to ensure that the experiment is done properly. In some cases, it is talking about things that are limiting factor to the experiment, that those that you can't control. So there's this controversy. I'm not too sure how you will explain it, but this is how we did. There's still this unresolved point among many teachers as to what precaution really is. Now, we want to maintain that specific temperature to ensure that there is, of course, accurate results. So that's a precaution. You want to ensure that what you're doing is right so that the result can be right. And we want to, of course, observe the experiment carefully to ensure an accurate count of the bubbles, or else we're not going to be having a good curve. So describe 2 precautions the student should take. So here it is something that the student should do to ensure that the experiment go as planned. So here they need to maintain that temperature, and of course they need to do that count properly. This was easy, I think. How could this experiment be modified to investigate the effect of pH on enzyme activity? Now you are modifying this experiment pretty much to see how varying pH would affect the rate of enzyme activity. So the first thing that we need to do, we need to use varying pH in each of the 7 test tubes. Rather than using different temperature, we're going to be using different pH. Now another thing we want to do is to maintain the temperature somewhere around room temperature for all 7 test tubes. All but temperature from the previous experiment is maintained. So we're going to keep the crushed Irish potato. We're going to keep the 1% hydrogen peroxide solution. We're going to put all this in the mixture. We're just going to change temperature, and in this case we're going to be putting in pH and varying pH. We're going to use a pH meter or pH paper to test the pH of each tube, and of course we want to ensure that we count the number of bubbles for each test tube. Pretty much the same, like I said, just to change out that which is of course temperature into pH, and to maintain temperature for the same for all these test tubes, and you would have manipulated the experiment for pH. Now we're supposed to name 2 enzymes that function in protein digestion, and of course describe the role of each. Now for CXC, 3 enzymes are going to come to mind very quickly. The first is going to be pepsin. Everybody will say pepsin. The second is going to be renin. Everybody will say renin. And the third is going to be trypsin. Most persons won't remember trypsin, as you're going to find trypsin being secreted in the pancreatic juice, and of course found in the small intestine. So pepsin is found in the stomach. It's pretty much an acidic environment, and it breaks down protein into peptones. Not protein to amino acid, but instead protein into peptones. Then you are going to have trypsin that is secreted by the pancreas, and it works best in a basic environment, and it converts those peptones that were broken down by the pepsin into polypeptides. So there we would have answered the question. But there is also renin, and you can tell me in the chat, what is it that renin do as an enzyme acting on protein? Leave that in the comment section. That's for you to show that you have been following the paper here, and you are pretty much a part of what we do. So we're looking at the other question here, C of question number 1. Before food can be chemically digested by enzyme, it must first be mechanically broken down in the alimentary canal. Now here you're supposed to name 2 parts of alimentary canal that are responsible for the mechanical digestion of the food. Now 2 parts, most persons will pop up very quickly. One, they're going to talk about the mouth and the teeth, and they're going to talk about the stomach, as the stomach is doing churning. But also as the food moves through the gut and peristalsis takes place, that is also going to help with some mechanical breaking down of the food, and of course pushing the food backward. So here we look at the mouth and the teeth in breaking down the food, and the stomach. Those are very important, but of course peristalsis would also serve a major role here. Now give 2 reasons why mechanical digestion of food is important. Now of course it increases the surface area for food particles to be chemically digested, or for chemical digestion. Another thing is that it helps to move food through the alimentary canal in a small, manageable portion. You really just can't take a dumpling if you were in Jamaica and just swallow that dumpling. You need to get it into smaller parts that it can move through the alimentary canal with great ease. The other thing that we're supposed to do, we're supposed to explain the significance of chemical digestion of food. So one would appreciate that in the mouth, as mechanical digestion takes place, chemical digestion is also taking place, where salivary amylase is acting on starch. And of course, we understand here that it is breaking down that mechanically processed food to release the chemicals. So chemical digestion increase the availability of food nutrient to the body. It breaks down macronutrients into smaller usable form for the body. We have to have an appreciation of that as to the importance of chemical digestion. That would have covered 25 marks, which is usually the amount of marks allotted for question number 1 on your biology paper 2 exam paper. You want to ensure that you have that all covered. Here we are looking at question number 2, 2AI. It has many sections, and in the box provided below, we're supposed to draw a food web consisting of the following organisms. So we know the algae here, and the small fish, and the tadpole, and the water snake, and the egret. Now, in doing this, there are some things that we want to do. We want to start identifying here the trophic level. So we know that algae is going to be 1, and we first trophic level. And when we are writing the trophic level, just let me show you that representation here. When we're writing the trophic level for the organism. So we probably represent this with a 1 and a little 0 at the top, like a 1 degree. So we want to label which one will come first, which is in the first trophic level, which is in the second trophic level, which is in the third trophic level, that type of a thing. You want to put that labeling beside these before you start constructing that food web. Now, having done that labeling there, which I was not able to do just now, you want to come on down here and start the construct. So what we know is that the algae, these are photosynthetic organism, and they're going to form the base of our food web. Then we are going to have the tadpole, which is of course a primary consumer, and this primary consumer will be feeding on the producer. Just let me show you the entire question here. Will be feeding on the producer. So you're seeing here algae coming from here being the producer, tadpole coming here being primary consumer, small fish is also a primary consumer, and then we have the water snake, which is of course getting some tadpole and some fish in, snacking, that is going to be a secondary consumer, and we have the egret pretty much doing the same. The apex predator here is going to be the eagle, and that would nicely top out this question, making it 3 marks easily we obtained just now. Now we want to know what is the role of the organism in at the start of the food web. Now this is very interesting here. When we talk about the role of the organism, we are pretty much looking at how it feeds. All right, so how does it produce or get food? So here the role of the organism at the start of a food web is going to be the producer. So it's making its own food, and it's making the food for other organisms and itself, whether it be in the food chain or the food web. So it is of course making food that is of course going to be passed on to other organisms in the food web. So particularly this organism, the algae here, is going to be a producer. 1 mark for that, and we move on down to the other question. Name the organism which perform each of the following function in the food web. I draw in AI, you'll see that. So let us look at what is happening here. For the first thing, we want to look at an organism that is both a predator and a prey, or a prey and a predator. Now, let us look at how is that represented in the food web. Now, here, to be a predator and a prey, it must have two arrows. So it must have an arrow leading to it and an arrow leading away. So this organism, the snake here, is a predator to the tadpole, while it is a prey to the egret. So you're seeing here it falls within the middle of two arrows. Now let us be reminded that herbivory, where this organism, the tadpole, is eating the algae, this is also a predatory relationship. It's called herbivory. So between plant and animal, that is also a predatory relationship. We want to bear that in mind. We're moving on down to the other question. So we chose here water snake and egret. So we're looking at, just let me get this again, I've lost my bearings. It is both a prey and a predator. So the snake, the water snake, is both a prey and a predator, and the egret is both a prey and a predator. So we just showed you two examples, but I think we were supposed to only do one, but we gave you a little extra there. A primary consumer, the tadpole is a primary consumer, and the small fish is a primary consumer. So we pretty much pulled that out there, primary consumer here, and we gave you these earlier. So let us look at the other thing that we have interest in here. We want to look at the third thing here. A secondary consumer is, of course, going to be the egret and, of course, the water snake. Remember, we were looking at that when we say primary, secondary, and then the tertiary consumer is going to be the eagle. And all that is nicely outlined here in your food web. Primary, this is going to be trophic level 1, trophic level 2, trophic level 3, trophic level 4. This is going to be a producer, primary consumer, secondary consumer, and of course, tertiary consumer. As we do that, we must bear in mind the different types of nutrition. The types of nutrition are autotrophic nutrition, echotrophic nutrition, and of course, saprophytic nutrition. Now, from the food web in AI above, we're supposed to draw a food chain consisting of 3 organisms. This is really easy here because it says 3 organisms. It could have said 3 trophic level, which would have been the same thing. So we have the algae, as all food chain must begin with a producer. We have the algae, a primary consumer here, and a secondary consumer. So this nicely tops off what food chain should be. Please be reminded of the organism that is doing the eating. But bear in mind that an organism could be a predator and a prey. So just show that flow of energy and be reminded that the flow of energy in a food web is unidirectional. Same for a food chain. Now, what is the role of the organism to which the arrow points